Vijay Kumar. Good morning. Vanakkam. Vanakkam.
Good morning. Vanakkam, sir. Vanakkam.
Thank you.
Madam,
inaugural start பண்ணிட்டாங்க. Sorry, inaugural welcome. Start பண்ணிட்டாங்க. என்னவா? நாங்களா இத நீங்க ரெஜிஸ்டர் பண்ணீங்களா?
A very good morning to all the excellencies, distinguished guests, participants, ladies and gentlemen. We are delighted to welcome you all to the International Conference on Advances in Biosciences and Biotechnology, ICABB-2021, organized by the Department of Biotechnology, Mother Teresa Women's University, Koregnath. A quote by Venter states that the future of a society 100% depends on scientific advance. And yes, that is absolutely true, and that is what our conference stands for. So without any further ado, may we begin this auspicious occasion with the Tamil Thai Vaithu.
Ask her to put play mode.
Middle button.
Middle.
Go to a full screen. Yeah. Looks like we lost. Nothing is playing.
வாசனை போல் அனைத்துலகும் இன்பமுற புகழ் மனக்க இருந்த பெரும் தமிழனங்கே உன் சீரிழமை திறமிழமிழ செயல்மறந்து வாழ்த்துதுமே.
Thank you all. Thank you, professors. Now, I would like to welcome the convener of the conference, Dr. A. Usha Rajnandani, ma'am, Associate Professor and Head, Department of Biotechnology, to welcome the virtual gathering. Please, ma'am.
Thank you, Uma. A very pleasant morning to one and all present in this virtual platform. Pavilion is made ready, warriors and knights get ready, but the victory comes by the grace of Lord Almighty. Exactly like that, a gift is given to us this auspicious day. We have gathered here for an international conference on advances in biosciences and biotechnology. Women are the largest untapped reservoir of talent in this world. We, as Mother Teresa Women's University stakeholders, feel proud in having a humble and great personality who have contributed a lot for our nation throughout her career. And Madam Dr. Vaidehi Vijay Kumar, our Honorable Vice Chancellor to this auspicious event. Hearty welcome to you, Madam. Next, in the midst of us, we have another dynamic personality. Everyone meeting her will be spellbound for her sweet words. I feel happy in welcoming Madam Dr. C. Hilda Devi, a Registrar of Mother Teresa Women's University. Welcome you, Madam. So it's a time to welcome our esteemed guest, Professor M.S. Reddy, founder and chairman of Asian PGPR Society for Sustainable Agriculture, consultant and entrepreneur, Auburn University of USA. Sir, you are actually a renowned personality in sustainable agriculture throughout the globe. By accepting our invitation itself, made our Indian scientific community feel great about this conference. We thank you for that and wholeheartedly on behalf of our university and organizing committee, we welcome you, sir. Welcome you, sir. So next we have with us the next resource person who is from Development and Evolutionary Genetic Laboratory, Development of Human Evolutionary Biology, Harvard University, Cambridge, who is going to handle our technical session, the second technical session. And we are really proud having you connected in the inaugural session itself. A warm welcome to you, sir. And here we have in midst of us Dr. Subramaniam Perumal, the zonal agronomist, Agrinos India Private Limited from Hyderabad, who is always supportive to our environment, our department activities, and stand beside us in helping all throughout our programs. A warm welcome to you also, sir.
Thank you, madam.
And once again, I welcome all. I also would like to welcome our professors, department faculty members, organizing committee members, the students, participants from throughout the globe, students, researchers, and faculty members of various institutions. I welcome you all. Once again, I welcome you all. Thank you for the opportunity. Thank you. Have a great day. Thank you.
Thank you so much for the warm words of welcome, ma'am. Michelle Obama once said, "We need all hands on the deck," and that means clearing hurdles for women and girls as they navigate careers in science and technology. Yes, ladies and gentlemen, at Mother Teresa Women's University, we are surrounded by wonderful women who put in lots of efforts to uplift women and girls in their careers. One such charismatic personality who works round the clock for the well-being of our university and the society is our Vice Chancellor, ma'am. I'm extremely honored to welcome her Honorable Vice Chancellor, Dr. Vaidehi Vijay Kumar, ma'am, to deliver the presidential address. Please, ma'am. Ma'am, mic is muted, ma'am.
Respected Professor Dr. M.S. Reddy, founder and chairman, Asian PGPR Society for Sustainable Agriculture, consultant and entrepreneur, Urban AL USA, and respected Chief Guest, Dr. Pushpanathan Development and Evolutionary Genetics Laboratory, Department of Human Evolutionary Biology, Harvard University, Cambridge, Massachusetts, US. Respected Registrar, Dr. Hilda Devi, respected Head of the Department of Biotechnology, Dr. A. Usha Rajanandini, and respected participants of this conference, other delegates, very warm good morning from Mother Teresa Women's University. It is indeed a great pleasure in being associated in International Conference on Advances in Biosciences and Biotechnology, ICABB 2021. Department of Biotechnology is organizing several events and they are working towards upliftment of the society, especially making women as entrepreneurs. One important product from biotechnology is the garlic, Malai Poondu, GI index for Malai Poondu, and which is definitely giving facelift for the farmers for getting more money on the product. And they are also doing the work on Karupatti Vellam so that this particular product becomes popular throughout the world. Apart from that, they have several other products from eucalyptus oil, how to prepare the room fresheners, and they have other important product on the line. So obviously, we know in the pandemic situation, the role of biosciences and biotechnology is very, very important. And this particular department will be in a position to collaborate with chemistry, physics in order to understand the molecular behavior, and the products are going to give you the strength for the human community to overcome this pandemic COVID situation. So apart from this, Mother Teresa Women's University offers several other courses which is bringing upliftment for the women. And we have 5 campuses in Tamil Nadu, one in Kodaikanal, another one Coimbatore, another one in Chennai, another one in Madurai. And incidentally, biotechnology, because of the laboratory, it is offered only in Kodaikanal. And the department has embraced biology also, and they are in a position to collect rare species in the plant so that these can be experimented all over the world. So lots of efforts are taken in order to get good score in NAC by providing several services to the women community, and we are organizing several such courses in order to prepare eucalyptus oil with the available plant in and around Kodaikanal. I wish the best for this department to have more credential, more patents, more products, more students, more paper publication, and be a role model department for the university. University kindly see to that collaborative research are taken up, and we are ready to send our students to your university, because Cambridge, Massachusetts, Harvard University is a very, very popular university, and Urban University in US is also one of the best university, and we are ready to have the strengthening of MOU, lots of other joint research, publication, collaborative researchers, and our students are hardworking students, motivated students, and they are ready to share their work experience. Faculty members are ready to share their work experience and the culture, whatever is happening in US and the culture of the work that is happening in Kodaikanal. We can have a balance so that our society can also be uplifted. Thank you very much for giving this opportunity. Thank you. Usha, Dr. Usha, am I audible?
Yes.
Ma'am,
Yes.
yes.
This is the souvenir prepared by
Yes, ma'am.
Department of Biotechnology, and I'm very, very happy to release this in the virtual mode. Thank you, Usha.
Thank you, ma'am. Ma'am.
Thank you.
Thank you, ma'am. Thank you so much for your talk today, and it's always as inspiring as it's always. And inspiring personalities always remind us to keep our heads, and standards high. Yes, ladies and gentlemen, among us today, we have an incredible person who has always inspired by her choice of words. May I now kindly invite the Registrar of Mother Teresa Women's University, Dr. Hilda Devi, ma'am, to kindly deliver the inaugural address. Please, ma'am.
Thank you. Respected and honored Chancellor Madam, the Teresa Women's University, Kodaikanal. Respected the Chief Guest, Professor, Founder and Chairman, Asian PGPR Society for Sustainable Agriculture, Consultant and Entrepreneur, Urban University, USA, and our another guest speaker, Dr. Pushpanathan Muthurulan, Development and Evolutionary Genetics Laboratory, Department of Human Evolutionary Biology, Harvard University, Cambridge, and the other technical special speakers from abroad and also national level and from Tamil Nadu, and our dear organizers and convener and the HOD, Dr. Usha Rajanandini, the
Nothing.
Dr. Usha,
Ma'am,
am I audible?
Thank you, ma'am. Thank you so much.
Dr. Usha?
சொல்லுங்க ஹில்டா, என்ன பிரச்சனை?
மேம் திடீர்னு எனக்கு அந்த இது கனெக்டிவிட்டி போகுது மேம். இதான் என்ன
मैंने ना पर करने वाला मैंने ना ऑपरेशन खत्म होना ना सब क्या आदमी मेरा चाहता है इंटरनेशनल कॉन्फ्रेंस ये हमारे पास है, ये मलेशिया से
Or Hilda, please ask the others to unmute, or administrator should unmute others, madam. Please.
हेलो
Hello, administrator should mute others except for the speakers. Please take care.
So as madam said, the biotechnology department have achieved a lot. As achievers, they keep achieving more and more. Now they have come up to the level of establishing biotech park, and they can also convert those inventions and also translate research into products and services by having necessary infrastructural support. So the biotech park offers needed facilities to researchers and small and medium-sized enterprises, entrepreneurs for technology incubation towards the commercial development of the biotechnology. I came
Thank you, ma'am. We are very sorry. We apologize for all the inconvenience. Please forgive us because we are in a hill station and morning we are having heavy rain here. So kindly apologize. Please bear with us for this. And now we would like to thank all the dignities, all the excellencies who have joined us today in the inaugural session. We thank them wholeheartedly. And now we shall move on to the session. To begin with the first session, we have an amazing personality with us today. I'm very happy to introduce him to you all. We have Dr. Professor M.S. Reddy, sir, and he is the founder of Asian PGPR Society. Today, we are very glad to have with us Professor M.S. Reddy, sir, and sir is the founder of the Asian PGPR Society, entrepreneur and consultant, Urban University, Alabama, USA. And sir is a professor in the Department of Entomology and Plant Pathology, Auburn University, USA. For 15 years, sir has served as a senior scientist and research and development global manager for several agrochemical industries, such as ExxonMobil, before switching to academia in 1998. Over 40 years, he orchestrated multidisciplinary global research for biological control of soil-borne diseases, biofertilizers, and biofungicides, induced systemic resistance, organic agriculture for safer, environmental friendly products, and poverty alleviation for rural farmers was a key objective. Sir has contributed over 350 peer-reviewed research papers and several book chapters. And Sir has worked with the national and international research and development organization funded by multi-million-dollar grants. National bodies in Europe, Australia, and many Asian countries regularly invite Sir to speak. And today it is one of our biggest opportunity that we have Sir with us. And Sir has trained several MSc, PhD graduates, postdoctoral graduates, and national and international scientists. And to add to Sir's crown, he has received prestigious international awards for research and entrepreneurship. And he's a successful business person, an investor, and an entrepreneur of Urban Venture. He consults for education's private sectors, government bodies, and NGOs globally. And more than this, personally, even now, I get goosebumps when I think that Professor M.S. Reddy sir is with us today. I don't know what a greater opportunity as a student I'll get from this. Sir, we are very, very happy to have you with us, sir. And we kindly request you to go on with your session, sir. Please, sir.
Please unmute. Professor sir, please unmute.
Thank you very much. Good morning to you all. Vanakkam, vanakkam to all my Tamilian friends. Is my screen is visible?
Yes, sir, it's visible, sir.
Yes, sir. Is it visible?
Yes, sir, very much.
Big screen, right? Hello?
Yes, sir. It's
Yes, sir. Full screen, sir. It's a sharing.
Yeah, okay. Thank you. Thank you very much. That good introduction. And my appreciation goes to our Vice Chancellor, the Registrar, and the in-charge head of the Department of the Biotechnology, and the faculty member of the department who corresponded with us very openly and promptly. That's why I'm here today. And also I have to acknowledge my friend for a long time, Dr. Subbu, who is the guy who wrote me back few months ago, sir, come back as a guest lecturer, the famous Mother Teresa Women's University of Kodaikanal of Tamil Nadu. Even though I'm born and raised in India, but I moved out from India almost 40 years ago. Still, I am so fond of Mother Teresa. With that name, today I'm here, even though it is midnight for me in the United States. My time is actually morning for you all. See here in this, I put all your administrators, Vice Chancellor, Madam Professor Vaidehi, and Dr. Hilda Devi of the Registrar, and your head of the department, Dr. Usha Raja Nanthini, and your faculty member of the department, Dr. Ilakiam. Thank you all. With that, me from the United States, I have to salute our Indian national flag, because I'm born and raised in India, or Indian soil, obviously Indian flag. And my university, who gave me bread and butter, Auburn University, Auburn, Alabama, United States. And as a founder and chairman of my own society called Asian PGPR Society. PGPR is nothing but plant growth promoting rhizobacteria. Based on this, my title today is the microbiome and gene editing, even though I'm not a gene editor, but because of your department of biotechnology and your topics for this event, I have to somehow use the term editing for future sustainable agriculture. I'm a born and raised an agriculture boy. I'm a farmer boy by birth, by training, by profession. Even now, I'm a farmer boy. So I will touch basically my experiences on this topic, because I'm not an expert in gene editing, but I'm so-called an expert in microbiome or PGPR. I will focus my talk today on the topic to produce, hopefully, for future rising populations in the world, not only India, not only United States, for global, in terms of sustainable agriculture. And I would like to see this form. I will touch, and also I will touch why I am fond of women's abilities to get education, irrespective of their levels of finances. I will touch that topic today very prominently, why woman has to be empowered for future sustainability, or a future food production, or a future safe and healthy food for the rising population around the globe. With that, all the administrators, madam, thank you for a very great welcome to me tonight, for me, morning for you all. Thank you very much as a first speaker for this event. The next slide. I'll touch basically, based on my title, what is the microbiome? That's the term I used. Most of the participants may be students, scholars, PhD students, some faculty. Faculty may know this term. I would like to very basic terms, very basic. I'm not a big researcher, but I'm a basic farmer boy. I use all these basic terms in my talk today. Microbiome is nothing but microbiota. Means microorganisms such as bacteria, archaea, fungi, protists, algae, etc. And the theater of activity of this microbiota. The theater of activity is nothing but how this microbiota, proteins, peptides, lipids, polysaccharides, nucleic acids, their mobile electric elements, including viruses, phages, relic DNA, and their metabolites, and interact with the environment with the signaling molecules, toxins, organic molecules. Biome, a simple term, is a reasonably well-defined habitat which has a distinct biophysiochemical properties. Keep in mind these terms. Microbiome is the holistic approach. For example, for biotechnology students and scholars, this microbiome of human being based on genetics, surrounding environment, diet we consume on each and every day, and our lifestyle, and the hormones in our body, and industry who could promote this microbiome of the human being. For example, nose has a microbiome, mouth has its own microbiome, lungs has its own microbiome, stomach, colon, and sexual organs, whether female or man, and our skin around our body do have several microbiomes, means microbiota or microorganisms. With that human, I'm not a human scientist or a human microbiome scientist, but I do know as a professor, I can learn, I can teach, I can enlighten people, whoever they may be, such as you. My main interest is microorganisms. You all know microorganisms, the tiny organisms living in all kinds of environments. Microbiota is nothing but a community of microorganisms in a specific environment. Microbiome is a community of microorganisms and their role within a specific environment, considering environmental conditions and interactions with each other. Today, the microbiome, the topic I have chosen for you to enlighten what it is all about. And also I mentioned gene editing in my title. What is the gene editing? What is the gene editing? A buzzword these days. Gene editing is the process of making a tiny controlled change in the DNA of a living being to produce GMO, genetically modified organism, by several ways. And in DNA editing, today I'm using a technique called CRISPR/Cas9, an enzyme called Cas9. CRISPR is nothing but clustered, regularly interspaced short palindromic repeats. The term makes a reference to a series of repetitive patterns in the DNA of bacteria or archaea or fungi or any microbe or in a plant. How this CRISPR technique will work? Just like cut and paste. Use a scissor to cut a DNA, a fragment of DNA. Take that fragment. Just for example, everybody knows a cell. A cell, either maybe a plant cell or a human cell. Every plant cell or any plant cell has a nucleus. That nucleus has a chromosome. We all know. That chromosomes have minute particles, shining particles. Those particles, nothing but DNA particles. If you take a scissor, cut a fraction of the DNA, which is I call a guide molecule, with Cas9 enzyme act as a scissor to cut the DNA segment. And you can take out bad segments, use the good segments, which I mean healthy DNA strand, put into a given microbe or into a plant to express what that particular healthy DNA strand exhibits. That's called DNA editing. If that many people in these days calling GMOs, genetically modified organisms, or the new technology, CRISPR gene editing, will touch GMO crops, GMO vegetables or plants, and CRISPR gene edited, inserted, and then growing from those technology, maybe a vegetable, maybe a crop, maybe a cereal crop or a fruit crop, will touch basically these terms in terms of research for future sustainable agriculture. Before that, how this microbiome technology could contribute for the future sustainability in food supply and industrialized and urbanized societies, clean environment, climate change and mitigation, healthy lifestyles. And we want to eradicate no poverty, we want to take out no poverty, zero hunger, decent work and economic growth, sustainable cities and communities such as Chennai, Kodaikanal, Calcutta, Mother Teresa's favorite place, and has to be responsible consumption and production using these buzzwords, clean water and sanitation, decent work and growth, economic growth, decent growth, decent work and economic growth for the biotechnology students and scholars for the future. Life below water, life on land, affordable clean energy. We don't want to use any carbon to produce energy. Decent work and so I mentioned that. Climatic action, good health and well-being. Good health, everybody has a lifestyle to go to the gym. These days, there are well-being, healthy styles, gyms all over the globe, including India, including Kodaikanal or Chennai, everywhere you see a good gym. Now they call lifestyle health clinics. How this technology could influence those. Let's put up some set of goals for microbiome for future sustainability. As I mentioned, word called no poverty in the future, hopefully, zero hunger in the future, good health and well-being for the future, quality education, gender equality will touch basically women and the men. We have to take out that disparity between the sex, between the men and women. Clean water and sanitation, of course, everybody want to drink good water, maybe a bottled water, maybe a tap water, maybe a river water, maybe a well water, may not sea water, of course, salted. Affordable and clean energy in your homes, offices, or universities. Decent work and economic growth. We need more money for daily living. Industry, innovation, and infrastructure. For example, your office is a closed room or outside your university campus. How the atmosphere is, whether it's clean, polluted, how we can change that polluted atmosphere around you. And we would like to have sustainable cities and communities around us. And responsible consumption, production. What are we eating every day? Of course, carbon monoxide going out, carbon dioxide going out in the atmosphere. How to control that? Climate action. Life below water. Aquatic. Life on land. We are living on land right now, not below the water. Only swimmers, fish or something else, fauna. They may live below water. We as amphibians, we as human beings live on land. And we need a peace within us, around the globe. Peace, justice, protect human rights. Dr. Ambedkar's Indian Charter, Indian Constitution, order. We need a justice for each and every one, not only in India, not only in Mother Teresa University, but around the globe. I'm a global man, so I would like to see that global justice. And we would like to bond strong institution, just as Mother Teresa's University have several campuses within Tamil Nadu. And partnership for the goals, your Vice Chancellor mentioned, she would like to have a MOUs with the university such as ours, Cambridge, and other university around the globe. Madam, Professor, Vice Chancellor, you keep that in your goal of your mission for your Mother Teresa University to have your sustainable goals for your university having international collaborations. Just a few examples. The term I used, the CRISPR-Cas9 technique. Here is a human body. Human means our body. You take out some blood from my body. If I have some problems, I call it patient body. T cells, take them out and use the technique CRISPR-Cas9 and grow those cells in the lab using, I said, cut and paste, cutting like a scissor with a Cas9 means then you introduce that CRISPR, then I call CRISPR edited cells, grow them millions of these CRISPR edited T cells in the lab. and put them back into human cells by this technique means putting a good DNA fragment into the human body. If so, it has to be characterized and identified what this fragment of DNA inserted into T cell could do. That's the one way of doing. And also you all see COVID vaccines. I am not sure they use this technology to produce the vaccines for against COVID virus, COVID pandemic disease symptoms. And another example, classic example, easy example I have chosen, but this is not my research. This is a borrowed research from somebody else. How to stop a heart disease in embryo or a newly born child or a growing adult, maybe a man or woman. For example, take a male sperm from a man, of course, man produces sperms, with a mutated gene sequence and the CRISPR-Cas9 inject into egg cell produced by female. The CRISPR-Cas9 into the sperm has that good fragment putting into egg cell of a female, and then female has a female DNA. Then the Cas9 cuts to male DNA in newly fertilized embryo. The sperm with the egg and embryo, this is the one. Then a small gRNA molecule guides Cas9 scissors to damage the sequence of DNA to make cut. The diagrammatic way. And the mutated gene sequence cut from male DNA is replaced with a healthy gene from female DNA. And the 72, by doing that, 72% success. Gene editing resulted in 42 out of 58 embryos with a 2 mutation-free copies of gene every cell. By doing that, they stopped the heart disease. That's one way of doing it. How an embryo can be edited to prevent HIV. You all know what is HIV. Again, a sperm and a healthy egg put together. Then you will give an embryo. Then use this CRISPR technology to cut out the bad DNA and insert it in the embryo. This knocks out a gene called CCR5, which creates the doorway protein that lets HIV into human cells. By doing that, we can prevent HIVs, whoever may have, by doing whatever, unwanted, whatever sex and whatever you may call. So we can knock out those HIV symptoms in that particular patients. That's a human style, but I'm not a human scientist. I don't do this kind of research. It is a borrowed techniques. I'm just introducing for understanding purpose only. I have not done any research. I have not published anything. As a professor like you all, or scholars can borrow, can explain in a given class. I am more interested. I'm a plant microbiome scientist. All my life, for the last 40 years, I devoted plant microbiome, nothing but PGPRs, plant growth promoting rhizobacteria, minute microorganisms. This is a so-called healthy leaves. Here is how this CRISPR-Cas editing will work. Mentioned, we're talking about DNA fragment. Generally, in the natural way, DNA breaks and also get it repaired. It's a natural God-given gift. But using this technique, we can alter genetic and physical attribute expressed by DNA particles or DNA fragments. In a given plant, this technology, CRISPR-Cas, reads the DNA of a plant. It may delete unwanted, it may edit wanted, and it replace an unwanted DNA fragment in that given plant. If so, as a farmer, and also I'm a consumer, I eat every day some food, breakfast, lunch, and dinner. Whatever I use, whatever I consume, has to be better nutrition. Has to have a long shelf life. Whether you put on the table or on the kitchen counter, or you may put in the fridge, or you may freeze it in a freezer. And it has to be disease resistance if you grow them outwards, has to be drought tolerant, and it has to give good and healthy yield. We're talking about now agriculture here. I am looking healthy plant gives a healthy food for future consumption of the rising population. In a conventional plant breeding, you know what is a plant breeding for plant scientists? You need a multiple cycles of breeding over several years to get a variety that give a good yield. By using gene editing, introducing CRISPR-Cas technique, we can reduce those number of years, maybe 1 year and 2 years, we can produce a healthy, disease-resistant, high-yielding variety. And a normal conventional breeding, normal conventional growing a plant, it takes several years to get a good variety, cultivar rather, maybe 8 years. By using this DNA editing, we can cut down the span of that into maybe 5 years, maybe even less in the future. In 1 year, you can have a good variety of what you need. Having said that, the buzzword called GMO, genetically modified organism, or genetically modified plant, or genetically modified vegetable, genetically modified fruit crop, or fruit plant. Do we like genetically modified? As you all recall, several years ago, in Karnataka, Bangalore, by introduction of cotton GMO plant and other things, many Kentucky Fried Chicken restaurants burned because our Indian people do not like unwanted GMO modified crop growing in Indian soil. But I'm not I'm not saying today whether I'm against it or I'm favorable to GMO. Research is going on. Let it be that way. Let it be that way. Someday in the future. But in the United States, particularly in my own country here, is the largest producer of GMO crops, and we do not have a mandate to label them as a GMO food, unfortunately. But more than 30 countries in the world, they are against, including India, but the rules are getting eased off. They won't like GMO plants unless they label them very clearly. Whether you eat tomato or apple or whatever, they need to be labeled very clearly. This is a GMO, genetically modified plant produced yield, maybe a vegetable, fruit, or whatever, or that's called two buzzwords, GMOs or non-GMOs. But CRISPR edited plant is different than GMO. So take it that. Coming back is my own expertise. I do have at least some little. I leave most of the next 2 days, whatever you have seminar, speakers coming out, my great friends, my best friend, Dr. Srini Ramaswamy from Taiwan, born and raised in Tamil Nadu, is speaking. He is the best scientist, best entomologist in the world I know of. And my classmate buddy in my master's class from Andhra University, Voltaire, Andhra Pradesh right now. She will be talking. She's an IFS officer. So and also from Lethbridge Research Station of Canada, he's talking. And the next speaker is a famous speaker from Cambridge. And they will be more specialized than me. I'm only a farmer today. And I'm a very, very village-born guy. I don't know much of this biotechnology, much of this gene editing, much of this. But I do have a command to produce organically grown crop and healthy lifestyle, healthy food, healthy diet for the future. And I would like to develop more food for the hunger to the world, not only to Kodaikanal, not only Mother Teresa surrounded population. I'm now a global so-called little, small fish in a big ocean, just like that. And in a CRISPR context, I'm based my talk today for sustainable agriculture, basic microbial community, and their functional genes and circuits, and genetic and genome engineering, and something called recombinant single species in situ or in vivo or in vitro or in vivo, their functional genomics, and ultimately I may introduce the metabolomics, metagenomics, metatranscriptomics, buzzwords for biotechnology students, my friends. I am not expert in this. I do know these terms. I know what they are. My students, some of my students, very few, my postdocs, my international trainee scientists around the globe in my lab over the years, they have done some research in respect of my interest. However, because I have to go with the flow of these biotech terms to attract funding for my survival. I'm more specialized than the expert. I'm essay expert, biocontrol, biofertilization, biostimulation. That is my area of specialization. This is a complicated. I'm a PGPR man, plant growth promoting rhizobacteria, tiny cells, tiny microbes. I'm specialized. I know what they do, how to grow them, how to multiply them, how to select them good ones against the bad ones and put them back and onto seed, onto the roots, or spray onto them, or put them in the soil where we put our seed. That is my specialization. Here is an illustrated diagram above ground of a plant and below the ground of a plant. I call this rhizosphere. Leaves I call a pilosphere. Inside of this I call an endosphere. And we're concentrating. And again, within the rhizosphere, there is a rhizoplane. And within the stem or within the tissues called endosphere. And of course, the rhizosphere. So all these buzzwords below the ground and above the ground on the pilosphere, several microbes are there. They're colonized. They do their own activities depending on whether bad or good. Bads produce disease symptoms. Good ones fight against those disease symptom causing microbes. So I'm specialized in this PGPR and the PGPR characterization and their mode of action may be a direct action or indirect mechanism. They may produce indirectly HCN production, antibiotic production. They may do systemic resistance, produce hydrolytic enzymes, sideropode production, exopolysaccharide production. They do work in biocontrol. As a directly, they fight direct mechanism by nitrogen fixation from the atmosphere, phosphate solubilization, siderophore production, phytohormones, biofuel production, root colonization, ACC deaminase production below the ground in the rhizosphere or rhizoplane. All these by these PGPRs, good bugs, good microbes. And then we all already introduced gene editing. It may be a metagenomics or DNA sequence, which is general, and metatranscriptomics, metaproteomics, metabolomics for microbiology, biotechnology students. So you keep these words one in your mind, taking classes and then synthesizing them, using them in your future research. That has a lot of future. I hope so. I believe in. In a given plant ecosystem, we need to worry about the ecosystem sustainability. And my main thing is agricultural sustainability or farmer sustainability, because I'm a farmer boy, as I keep on mentioning. And we need to take care of the soil. We need to take care of the given plant, and with some genetic engineering. However, my main interest is microbes, and my interest is the soil. So I'm concentrating on microbes in soil, on soil, below the ground, above the ground, to improve soil structure, organic matter, soil aeration, soil buffering capacity, balanced nutrients, soil fertility, to improve the growing root and the shoot, and ultimately growth and yield on any given plant or any given crops, maybe veggie, maybe horticultural plant or fruit, or row crops such as sorghum, bajra, maize, corn, and finger millets, whatever you may call, on those ag veggie crops. So this is a diagrammatic plant. And I'm introducing PGPRs, plant growth promoting rhizobacteria or plant growth promoting microbes. whether alone or the combinations. I call a consortium. I would like to protect this plant by abiotic stresses or from biotic stresses. You all know stresses in a given environment on a given crop. It may be a drought, no water, water stress, flooding, heavy dose, heavy rain. These days I've seen all over India, heavy rains, flooding. Several years ago, Chennai flooded. Sometimes extreme heat, I think 100 plus. Foreign heat, maybe 50 degrees Celsius. Some areas in India. Of course, we don't have freezing other than in the Himalayas, or particularly for us in the United States in several areas. And some soils don't have nutrient stress. So these biotic and abiotic stresses on a given plant, what we could do, how we can remediate these stresses by introducing plant growth promoting microbes in the system. Coming back to sustainable agriculture, with the microbial community, we've been talking on, my topic is the microbiome, particularly plant microbiome. With this plant microbiome below the ground and the above the ground for sustainable agriculture, we may use directly microbes to fight or work as a biocontrol, or to biofertilization or biostimulation. That's a normal way of doing it. Thus, I like it, this way. But gene editing, gene safety, and genetic or genome engineering, recombinant technologies, and metabolomics, metagenomics, metatranscriptomics. That's a trend in biotechnology today. I would like to have both together, plant microbiome with new technologies. Can we have more effect or more influence, more efficacy? Again, I'm keep on showing this plant. My plant microbiome would like to breed and for a new plant, the new microbiome, to optimize and customize the beneficial effects in a plant through plant microbe interactions. And we would like to have friendly plants. And I am more into organic amendments, soil amendments such as organic, inorganic, and others such as manure, crops, residues, compost, agricultural residues, and perlite, vermiculite, lime, sand, macro fertilizers and chemicals and fumigations. I don't like chemicals, I'm anti-fumigant. And basically, I'm a biological pro-soil crust scientist. I would like to have a healthy soil for a healthy plant. When I put a seed in that soil, it has to germinate, it has to produce a good root system, and eventually produce a green shoot system and give a good yield. To do that, we would like to apply these microbes as a seed treatment, putting into the soil, inject into this, or spray onto this philosphere, or on the leaves, or on the whole plant, to protect unwanted biotic and abiotic stresses of plant diseases or a pests. So we can grow healthy plant for a healthy yield to produce more. Again, same thing. Given a plant, just this particular, I am more concentrated, more interest to look after below the ground, around the root system, the surrounding called rhizosphere. In a normal plant, you throw a lot of fertilizers in for a given rice plant. It give a healthy rice plant, gives a lot of yield. It doesn't matter what is healthy or not. If you need a more yield, you get more by throwing a lot of fertilizers. No, we still need those, but I'm more concentrating in a nano fertilizers, nano, micro, macro fertilizers has no residues in the soil in a given root or a yield. So we will be more focusing towards the macro, macro or micro based nanoparticles. If you want, is the conventional fertilizers. But if I put my microbes with a nano-based formulations with those macro, low dose of macro fertilizers with the combination of my PGPRs, plant growth promoting microbes, we can have a bumper healthy plants and ultimately yield. Just move on this. So again, the plant microbe. So keep on focusing below the ground on microbe, philosophical microbe, or called endophytic microbe. Ultimately, soil microbes, the rhizosphere microbes are very important for any given plant to grow them healthy. But in a society where we live in, whether it may be India or United States, Canada or Europe or South America or whatever may be a continent, the people around us, they have to like it. Society, it is profitable, is the economy by using these technologies, and are they compatible to the environment? So we need to keep in, we need we do to grow a plant to produce food. So this society has to approve, means these things has to be compatible, these societies, and have to be profitable, return on investment on this, the so-called complicated plant microbiota and immunity and a chemical communication between plants and microbiome, and the environmental variation. Every day I said biotic, abatic stresses, and heavy rain, no water. So we need to keep in touch these two societies or two environment, plant environment, outside environment, below the ground, above the ground, whatever it may be. We need to keep in touch. It's very hard. It takes several hours to explain each term, but it's a general phenomena by a diagrammatic way. Again, back to the bench, lab, greenhouse and field research, the new technologies, gene editing or CRISPR-Cas9 editing, they can put in any plant or on a leaf or by growing them using a tissue culture technology, a lot of test tubes, a lot of tubes, a lot of petri dishes. This technology, the people keep on working and over and years and years. Sophisticated new results coming out by using this CRISPR-Cas9 technology. Those technologies are very well advanced, growing into the greenhouse and also field. This kind of technology, the CRISPR-Cas9 technology, and a conventional microbiome technology, introducing those microbes, PGPRs, in any given ornamental row crops and fruits and veggies, and particularly in vertical way of growing. You see in India, every rooftop has a vertical plants. Every rooftop has people growing their own veggies. I call them vertical agriculture, vertical development. You just need stuff. You can grow any vegetables using these microbes, beneficial microbes. And this conventional way, throwing as much as you can, the fertilizers, chemical, herbicide, pesticides, insecticides, all kind of toxic stuff. I'm anti those kind of stuff. I'm very favorable my microbes and semi-favorable this gene editing technology. But we have to go with the new technologies. I don't know. Otherwise, I will be very old orthodox scientist. Again, in the conventional way, using microbes. This is my tiny plant. I'm more interested below the ground. All very careful. The root rises per interaction and introducing maybe a single, multi, or a combination of microbes in the system. If so, those will colonize this growing root system by doing producing anti-compounds and colonizing growing root system and giving good root exudates and then absorbed by the growing root system. And if so works, I would like to see, for example, this is rice paddy field. I love it. And by introducing these technologies, we could grow healthy, beautiful rice fields. Then ultimately it may be a basmati rice, it may be Madras rice, sticky rice kind of style, or whatever it may be. We can do several things. It is a classic example. People have advanced technology. I am more orthodox, conventional, plus organic. My organic tomatoes grow like this with my microbes, my compost. If I use gene editing technology, those tomatoes can do these kind of miracles. This is a CRISPR gene edited tomato versus my conventional PGPR produced tomato, this tomato, that tomato, these. By using good resistant varieties, growing with the microbes in an organic way. But when you compare with the gene edited plants, they produce enormous amount of yield. They may be tiny, they may be very tasty, they may be very good texture, they can store a long time compared to these conventional plant breeding varieties are growing in organic way. But I love it both way. I love this technology. But I do believe what I learned, where I born and raised in normal way. But I do use the compost. I use the organic way. I'm a vermicompost guy, earthworms, and a compost together using beneficial PGPRs. I love to do that way, and I love to eat that kind of food rather than, for time being, I have not consumed any genetically modified or CRISPR edited produced veggie or fruit or anything so far. As you all know, the richest man, Bill Gates, is buying each and every acre available in the United States because he's the richest man. He has a lot of money, millions of acres, introducing the CRISPR-Cas gene-edited technology. He would like to develop all in his acreages with a sophisticated technology to produce disease-resistant, high-yielding varieties. Started with the corn right now. That is his way of future. He's a Microsoft guy, so has the money. So he wants to invest into agriculture, revolutionize this agriculture for future. Whether you like it or not, because he's a rich man. Anyway, so far we're talking healthy plants for a healthy planet. Plant health is your health, right? If you eat a healthy food, your health will be all right. And how do we feed the world hunger? I don't want to go through this, but you all know McDonald's, Kentucky Fried, and all kind of restaurants and drive-through restaurants, fast food restaurants. instead of using a hamburger, it may be a chicken burger, as a goat burger, a beef burger. Of course, people don't eat in India, but here in the United States, rest of the world, many people, majority they consume the beef. I'm strictly Indianized, but rather eating either chicken or fish or goat or whatever, I rather to have a veggie burger. Veggie burger. It may be a tomato, it may be a bean burger, it may be a carrot burger, it may be something else. I would like to see these veggie burgers coming from organically grown produce. So as I mentioned, my topic is a sustainable agriculture, whether in India or outside. And there's a new buzzword, new technology for the future called regenerative farming, regenerative agriculture. Bear in mind, regenerative agriculture is nothing but use the growing practices that improve the health of their land. Methods include the increasing biodiversity, rotate the crops, minimizing chemical inputs, toxic stuffs, no-till, and don't clean your land. Use them and stay up, leave them there to decompose and giving organic. Integrating livestock, if you see older agriculture, buffaloes and cows and goats and sheep, all kind of stuff, they take to them to the field and whatever they need to excrete. So is healthy to the growing crop. So that means we need to integrate livestock and use the cover crops. All these are traditional, conventional, modernized, organic way is called regenerative agriculture to enhance the carbon accumulation in the soil, to make the soil to be rich. Just ignore the same thing, so keep in mind what I explained before. Again, same, I mentioned, just take us as a human body. If I am here, if this is my body from top to bottom, I rather use to be healthy, right? Rather to sick. If we want to be sick, you want to go on conventional food, rich in meat, low in fresh fruit, low in solids, pesticide residues, poor microbe grown food, conventional way herbicide, fertilizer, pesticide, and these are all fragile health, even though produce. And if you use that versus a good health or by consuming sustainable agricultural produced food, biofertilizers, biostimulants, biocontrol microbes, or with my PGPRs, they may be rich in nutrients and low meat and rich in organic solids and fruits and raw vegetables. They may be pesticide-free, microbiome-rich, healthy gut. I mentioned human microbe. They may be very healthy. So we would like to focus on good health. From now on, I'll go very easy. And these, as I've been mentioned, PGPR, good microbes, plant microbiome. These are my own research produced microbes over for the last 40 years. I lived in Canada for 15 years, graduated from British Columbia, Canada, then worked for Exxon into several industries in Canada before I moved to United States as a professor. This is my Canadian product for a forest industry to produce healthy silviculture by using PGPR in liquid formulation, introduced as a seed treatment, nurseries and fields, and reforestations, and to produce healthy forest plants. just an example. Then I worked on peanuts and cotton and used nanoparticle products, developed a tremendous high efficacious nano-based products called, one is called optimized lift for peanuts and bolt for cotton. As I mentioned, I'm not just an ordinary scientist to publish 350 papers, books and stuff, but I do use them how to make use of commercializing the research. That's why giving an example, some of my PGPRs for rice and paddy and soybeans and cottons and tomatoes, and with several formulations, powder, particles and capsules and liquids. We have done a lot of research for the last 40 years, not by myself, my students, my postdocs, my international scientists, and international collaborations. I'll come back to that with the MOUs in national, international, multinationals, with the several scientists, not just by myself. To put together synopsis of my products, just few. These are commercial, they're available in any country, but it's not by myself. I'm only researching, provide these products with several companies around the globe. They've been marketing by these trade names to various crops to make their own money, because they invested their research funds in my lab, in my university, with my students, my postdocs, and my scientists. With our technologies, we have given them our sophisticated products to them, because we're not sophisticated in fermentation, formulation, packaging, but we do give them protocols that they're responsible by those industries, just like Dr. Subbu's company. But I have not worked with them, but I do know them, Agrinos, and now a new company they have merged with. So these are, anything from any crop or veggie or fruit or row crops, we have commercialized our research with through new innovations. Having said that, having done research for the last 40 years, so many techniques, so many technologies and methods, with so many students, postdocs, and scientists around the globe, I mentioned I'm still a farmer. This is my backyard here in the United States, behind my house. I do grow my own veggies and the flowers and all kind of stuff. So just showing, using my own techniques, my own backyard grown microbes, compost, organic, no fertilizers, no chemical, no pesticide, no insecticide. This is a beautiful, my backyard garden right now live. Anybody next time visit me in the United States, come and visit. It's a mini farm, mini beautiful, sustainable backyard in my home. So I use my techniques. I'm just showing because I don't just believe in my research publication. I rather do every day because I can see them every day, how they're doing it. There are a lot of veggies, a lot of my curry leaf, my whatever, my wife and coriander and eggplant, so many I have in my backyard. They're very healthy and keep producing. And also see the yield. Every other day we have to grow this Indian bean, chikudu, Telugu in Andhra. I love it. I love it. My, Michika Bhaji, famous, I love them. This jumbo pepper, plenty. I have several pounds and kilograms coming out from my plants, and we supply to many my communities, my friends and local farmers, and whoever needed, they come and pick them up. No charge, you pick, you come and you pick, you pack, you take home and eat it. apples and okra, bitter melon, eggplant, tomatoes, peaches, and other squash, beans, whatever I have. Okay, having that experience in my own backyard, how do I disseminate to the people around the globe? That's why I started my own society called Asian PGPR Society for Sustainable Agriculture in 2009, registered in the United States and India. With this society, I have 1,500 plus life members from several Asian countries and other countries too. With this society, I've been advocating or teaching or giving lectures. Hey, use these technologies. For example, my colleague, professor here at Auburn University, is actually the father of PGPR. He's the man, Dr. Klopper, he's the man founded the word called PGPR. I call him father of PGPR. He's my colleague. We work together in the same university. So I have to show him the father of PGPR, the guy who coined this term when he was a graduate student doing his PhD in California in the United States. And him and I, prior I started PGPR Society, we started international PGPR workshops. We call workshop. We go every country, every continent and teach with a bunch of scientists and the students and postdocs and progressive farmers. We started that workshop in 1987 in Canada. Our aim was to go around the globe every 2 years, every 2 years, and every continent in the globe, take our technology of PGPR and the science and the technology and the associated techniques, go and teach them around the globe. Since 1987 up to now, 11 workshops we conducted, time 3 years, 33 years without a gap. We have covered many continents, Canada, Switzerland, Australia, Japan, Argentina. I brought all my group to Kerala, India too. And then Europe, Netherlands, and United States, South America, Belgium, and then back again into Canada. So we covered entire continent. Also, we went to Australia, except South America and Russia. 2 continents left under these workshops, but then we thought it's a global phenomenon, man, a lot of money, a lot of effort. We have to spend our money, our government money, university money, and grants money to go all over these conferences, bringing people. It costs them to come and travel and listen to us. So we thought of, Dr. Klopper and I split it. He said he will take care of Latin America. Then he asked me, can I take care of Asia? Because I'm Asia. My blood is still from Asia, of course, from India. Even though I learn, whatever I learn, I teach for bread and butter. My brain and my Atma, my soul, lies with our Asian people, particularly India. Friends, I'll never forget, our Indian soil gave me birth, of course, mom. And of course, I call Matrubhumi. So I never forget, born, raised, educated up to master's level. I have to reciprocate the Indian soil spent on my education in the early days until I'm adult. So with that, I have chosen, I took a challenge, how I can reciprocate the India investment in me. Having said that, with that Asian PGPR Society, we started in my own combined state of Andhra Pradesh in 2009 in Hyderabad, beautiful international conference. Then my aim is every 2 years, I would like to set up these conferences in every Asian country. Having said that, our country, India, started in Hyderabad, then moved 2 years later, Beijing, China, Manila, Philippines, Hanoi, Vietnam, Bogor, Indonesia, 3 years ago, Tashkent. We thought of how 2 years later in Malaysia. This is a bad pandemic. Whoever created this damn stupid COVID stuff, we stuck. We could not do it. What we're planning to do next year in Kuala Lumpur, Malaysia, is not only me, my friends and folks. I have an extended family like you. India is big family, but my scientific community is my extended family, folks. Hyderabad folks, Beijing folks, Manila, Vietnam, all these folks. Having those extended family, I am starting in each and every Asian country, their own chapter of Asian PGPR. So far, 8 countries of Asian countries started their local Asian PGPR, started by, of course, India, and then Saudi Arabia, Malaysia, Indonesia. That's why we're having a Bali conference end of this year. And Uzbekistan, we had 3 years ago. And Pakistan, they have started. And Turkey, they have started. And Sri Lanka, I've been there. All these countries I've been there. And I'm promoting, hey, you have your own country chapter of Global Asian PGPR Society. So more localized, just showing for your interest, and particularly my heart is there with them. Coming up, 2, 3 conferences. Number 1, immediately end of this month, virtual conference in Bali coming up. Please join us. The 250 people registered. I'm bringing my close friend, Dr. William Dodd, spent 15 years in ICABB, Hyderabad, as a Director General, now is the Agriculture Minister in Philippines. Good friend of mine I invited him. He's going to give his message to the Bali conference along the Bali governor. Of course, I will be virtually participating. Please join us for that. Then immediately next month, September 1st week, we're having the Indian chapter conference in Bhopal, Madhya Pradesh. Those folks of Indian chapter organizing, please join that conference also, all virtual. If at all COVID is good, travel conditions ease off, and I will be monitoring WHO notices to travel. Please, if so permittable, your travel funds permits, your university or funded company can provide some money, come over, very cheap registration fee. Next year, August, in Kuala Lumpur, Malaysia, International 7th Asian PGPR International Conference. Now here I would like to bring Mother Teresa Women's University mandate. You may have your own mandate, you may have your own mission, but I am an advocate of reach to unreach, and women empowerment. With a woman in globally, any country, anywhere, in a house, office, university, industry, without women empowerment, nothing can prosper. I think Mother Teresa University mandates, I have read about your mandate, where Mother Teresa came from when she was 12 years, decided to serve the people, without any discrimination, poor or rich, hunger and stuff like that. When she became 18 years old, she took oath became a nun, spent all life until she died in Calcutta. I never met her personally, and I have glanced through her dedications and glanced through her Nobel Peace Prize and whatever. And starting this university, honor and name, not only in Kodaikanal, I think you have 4 or 5, if I recall, several other centers, extension research station. Ma'am, Vice Chancellor, Registrar, and Head of the Department of Biotechnology, and a faculty member, and all other faculty members, irrespective of the department. I still believe in the famous leader, my famous man, Sri Honorable Narendra Modi ji of Prime Minister of India, global stalwart, has given a mandate in International Women's Day. I'm open. Any woman call me, have a question for women empowerment. Not only Modi ji, though I'm a favorite, I'm not a politician. It doesn't matter what party he belongs to. I believe in India. I believe in his administration. And I believe in his future mission to India to become a powerful nation. We're already there. We're already reaching 3rd in the world, I think 3rd or 4th in the ranking. I think in a very short time, the India have overcome the so-called rich countries such as the United States, China, or something else. I'm hoping for God blesses Modi ji, God blesses India. And of course, every queen or, the Bill Gates' wife, Melinda, what is happening, the family life, Mr. Bill Gates and Melinda Gates. That is their personal business. They're already separated. That's okay. But both have their own trust, helping. And you mentioned, Madam Vice Chancellor, Madam Registrar, you mentioned the ladies' empowerment. You need to introduce self-employment by those women students, girls or adult girls or women, whatever the profession. This is a cosmetic profession. And there's a Rangeli profession, produce an art for, whatever festival, they throw the kumkuma, throw the colors and stuff. And they control their own markets and everything. Of course, because the girls and women procession. Hey, we need a power. Man, guys, give us more respect. Give us more money for wages. And give us more salary than men. Stuff like that. I believe in it. I want to empower the rural women, particularly the key for the ending hunger. So with that, but this bring back the short bio-based technology, PGPR-based technology, the ultimate goal to revolutionize in plant breeding to produce more food. Here, our population will be rising in 2050 to 10 billion people, almost 10 billion. So having these technologies, PGPR-based technology or CRISPR-Cas9 technology, gene editing or genetically modified, we would like to use a conventional way and a sophisticated robotic technologies, robots, and digital farming, all together, integrated approaches. That's why I'm showing to feed the growing 10 billion population very shortly. Now, the last but least, me, as I mentioned, going around the globe, many continents, many countries. So far, I visited 50 plus countries to advocate, to give seminars, to stimulate from student to researcher and professors faculty, particularly the administrators such as Vice Chancellors, unless they have a vision and mission, below staff may not be motivated. Madam Professor Vice Chancellor, I am giving you at least my idea. You be vocal, be focused, give as much respect to fellow students and faculties. Of course, you're doing, ma'am, on behalf of Mother Teresa. With that, to conclude, I would like to see whether it may be a Madras Thali, Andhra Thali, Kerala Thali, Delhi Thali, or Indian Thali. Andhra Mata, sorry, India Mata, sorry, I'm Andhra guy. Would like to Pancha Bhojanam, Navarasa Bhojanam, Kutia, whatever, Tamil, is maybe a Sambaro, whatever you call. With that, with this technology, to grow healthy, to get a good healthy diet for our daily consumption, with that, all, and see future forms with the digital, robotic, PGPR, CRISPR-Cas technology in giving these future forms. With that, I salute our national flag and, of course, my bread and butter country, United States. I have to salute because made me today what I am. Of course, Vanakkam to all Tamilians. Namaskar to rest of country folks participating in today's event. God bless India. God bless Mother Teresa Women University. God bless Asian PGPR Society. God bless the United States, my country. Thank you very much. Namaskar. Vanakkam. Thank you all. My pleasure. On behalf of your invitees, Madam Vice Chancellor, Madam Registrar, Madam in charge, Head of the Department of Biotechnology, Madam Faculty corresponded us, my friend, Dr. Subbu Perumal, and I inviting my future friends coming, Dr. Srin Ramaswamy and Subbarama Lakshmi, and from the Canadian speaker from Lethbridge Research Station, because I'm also a Canadian citizen. I'm a Canadian citizen, American citizen. Unfortunately, I don't have our Indian citizen. That's okay. I have multiple unlimited long-term visa. With that, if you have any questions, I'm very glad to take. Thank you. Thank you. So, Vanakkam again. Thank you all. Thank you very much.
Thank you so much, sir. And we would like to tell it in Tamil, Nandri, which means thank you from our side. That was a wonderful presentation, sir. Audience, if in case you have any queries, you can ask sir now. You can raise your hands to ask any queries.
Looks like no questions. Move on to our next speaker.
Ma'am.
Yes, you're audible. You can continue.
Good morning, sir.
Yes.
Sir, how does microbiome helps in analyzing forensics, sir?
Pardon me, can you repeat?
Hello?
Yeah, can you repeat your question, ma'am?
Sir, how does microbiome uses in forensic analysis, sir?
Forensic what?
Forensics.
Oh, not my, you ask those doctors coming up, next speaker, ask him that question. So I'm not a medical doctor. I don't know anything of forensic. But microbiome, don't use the microbiome in the forensic technology. Use the CRISPR-Cas technique to detect what you're looking for in that particular forensic lab to diagnose a particular specimen. So sorry, I'll ask the next speaker.
Thank you.
Thank you, Santana.
One more thing, I would like to give my open invitation. Please, anyone from your Mother Teresa University, young graduates, want to come to United States, any other country for future PhDs or masters or postdoctoral, but I do not have right now any funding because I'm completely out of research. But I can provide a strong recommendation to get you anywhere in the world if you're qualified, provided you have a TOEFL and GRE exams. Please use me as a because now I participated. I can write a good recommendation letters, rather reference letter to any country you select or anybody you select. But don't ask me to find a placement. You apply. When you go through your TOEFL or a GRE. Because foreign universities, institutions ask for 3 recommendation letters, right? Reference letters. Or Indian professor, you know how they write, how we write. So is the difference. So I can quote, hey, I have a collaboration with the Mother Teresa University. I know them. I can make up a story, not realistic. I will look for a resume. I'll look for your professors. Once I'm satisfied, I can write it. There's several thousands and thousands students across go benefited from me for their masters and PhDs, postdocs. Hundreds of them have got green cards. Now they became citizens in my country here in the United States. This is my open, telling you all, there's no secrets. God has given education from India. God has provided me enough. Human service is the best service, right? To reach to unreach. Unless there is somebody, it's very hard to come up. Anyway, go ahead. Any questions? So
Thank you so much, sir.
huh?
Actually, it takes a very big heart to tell that you wrote a recommendation letter.
I learned from Mother Teresa. She showed help we need. Be smile, even though some other person won't smile. You see her in our biography, a text, whatever she has 3 statements. Help someone in need. Smile even though they don't smile. Third one, keep society together. Don't despair them. God has given, I am doing right now. I don't go to temple. I don't go to coconut thing, that dandam, this kind of stuff. A fellow human being service, giving me boost. I am more than what I needed in this country. My children are highly educated. My daughter is a doctor. My son is an attorney. I have 4 grandchildren. Life, I cannot imagine. I cannot need more than what I need. Truly speaking. That service, my mom is illiterate. My dad is illiterate. My dad died when I was 3 months old. I don't know him. But a widowed, illiterate mom, because she's an agriculturist. Of course, I have 5 older brothers, all 5 brothers, no sisters, no nothing. That's why I have this women thing, reach to unreach, the women empowerment. My mom is empowered, a widow, illiterate woman, gave me education. So I have this in my brain. So I taught to my daughter, now taught to my son, now teaching to my grandkids. This is a family thing, but I'm teaching the world. God, whether Hanuman or Venkateswara Swami or this Swami, that Swami, I don't care. The human power, the power of power, nature inserted me. But now I'm more of this Chaya Wala, Prime Minister Modi ji, because I attended his Vaibhav Summit, invited me with him. There are things. I like your guy from Tamil Nadu, Subramania Swami. The MP guy, Twitter guy, whatever it may be. Some stuff. Anyway, I love all your chief minister. That's okay. That's politics. I'm not a politician. I'm a farmer boy. I taste my soil in my backyard. Amruta rasam. I squeeze my soil, squeeze the juice I put back into the plant with the compost, with the earthworms and some microbes in it. Kalusham. I don't know what Tamil called kalusham, Sanskrit word. So I think time is past. So go ahead. I think I given 11:30. Now is 12:30. I exceeded 5 minutes. Go ahead.
Yes, sir. Thank you, sir. It was not only brain-filling your presentation, but it also touched all our hearts. And especially when you said, I love this technology, but I still go for compost and biofertilizer. That is what is sustainable environment. You still have to work on technology, but at the same time, you need to think about compost and fertilizers. And when you said that scenery behind you is your backyard, oh my God,
Yes.
we're all amused,
Yeah.
sir. It is really wonderful. And I don't know, maybe we wouldn't get any better resource person than you, sir. In the beginning, we were very confident, everything was set, and having such a personality like MS Reddy, sir, for the international conference or on all the eminent speakers, we were a little overconfident what could go wrong. But then we found that, okay, we could have network issues and all that. But still, sir, out of all the thing, I could see the time that was going in your slide. It was midnight there for you. In spite of all your busy schedules and being midnight, you were there for us, sir. And all of us students, they had an idea about CRISPR, what is CRISPR technology and gene editing, and also about PGPR. And mostly we were very happy because we saw who was the father of PGPR. And that was very, very good, sir. Thank you so much for being with us, sir. And we are very sorry for all the inconvenience that happened before. And I guess you will be able to apologize, give us apologies for that. And thank you so much for being with us, sir. And more than that, I would also like to thank Subramaniam Perumal, sir, from Agrinos Private Limited, sir. Thank you for bringing such a personality. Maybe 100 times won't be enough for this, sir. I still have goosebumps. Thank you so much for joining us today, sir.
Excellent word. Subbu, he's a very, very passionate man. When I visited Hyderabad, he visited my house in Hyderabad 3 times just to meet with me. He's such a wonderful person. Subbu, I thank you very much. And of course, I introduced my colleague, my friend tomorrow coming. She's my classmate buddy in IFS. She's a passionate girl, lady now. You have to listen to her name. She's reach to unreached. She's more sophisticated than me because India, she's more into frequent contact than me here in the United States because of, color and racism, all kind of stuff. Thank you, Uma. Thank you, Uma, for you are very good. Looks like you have an excellent English.
Thank you, sir. Thank you so much.
Thank you. Thank you very much.
Thank you, sir. Thank you so much for spending your valuable time, sir. Thank you so much.
Thank you so much, sir. Thank you, Professor. We'll be in touch. You have my WhatsApp number. I have your WhatsApp number. Sir. And same thing you can extend to your Vice Chancellor, the Registrar, and your in-charge head, madam. Okay, not to students though. They came bombard stuff, but that's okay. Yeah. Thank you very much. I've taken this I have a good lunch for you. So let me sleep. And next 3-4 days, I have several other webinars.
Okay. Thank you, sir.
Thank you.
Thank you, sir.
Thank you, Doctor.
Yeah. I am signing off.
Yes, sir.
Sir, just one more minute, sir. Our HOD
Yeah.
wants to tell a few words, sir, and thank you for being with us today, sir. Just one more minute, sir.
Yeah.
Okay. Sir, thank you so much, sir. Sir,
Yes.
sir, we had a wonderful, it's night there and morning here, and it was great, sir, hearing from you. And you said last you help our students, and really they are also fortunate and blessed, sir. Thank you so much, sir.
Thank you, Dr. Usha. Vanakam, you go and eat the thali. I showed you. Lunch, right? You're going to eat nice thali, right?
Sir?
Usha, are you going to eat nice thali for your lunch?
Lunch.
Yes, sir. Yes, sir. Yes, sir.
Thank you. Thank you, Dr. Usha. Thank you. Thank you very much.
Thank you, sir.
Thank you so much, sir, and have a very good night, sir.
Yeah, thank you.
Thank you,
Take
sir.
care. Thank you all. Hope to see you very soon in Tamil Nadu. I have
Yes,
a lot
sir,
of
we are
friends,
waiting for that, sir.
lot of collaborations going on in Tamil Nadu. I've been several times. I'll come back.
Yes, sir. We are also waiting, sir.
Thank you so much, sir. Dear
Thank you,
participants,
sir.
we would like to continue with the session. Thank you, participants, for joining with us for the first session, and it was a wonderful session. And nevertheless, the second session is also going to be like that. To engage you in the second session, we have Dr. Pushpanathan, sir. He's a research associate at the Department of Human Biology at Harvard University, Cambridge, Massachusetts, USA. The overreaching goal of SIR's research is to use functional genomic approaches on understanding how the genetic changes and epigenetic reprogramming in the human genome contributes to genetic diseases. And in SIR's current research, SIR has identified 2 separate DNA mutations near the GDF5 gene in human, and one that can cause knee osteoarthritis in older adults and another that can cause hip dysplasia in babies. And SIR has completed his postdoctoral training at the Laboratory of Gene Regulation and Development and at National Institute of Health, Maryland, USA. SIR also has his postdoctoral work as he has developed a novel receptor based on GRASP method using CRISPR and high-resolution microscopy to map neurotransmitter receptors to brain neural circuits that involves visual motion processing in Drosophila. In his PhD work, Sir discovered a novel antifungal peptide, MMGP1, from marine metagenome using function-based metagenomic approaches and characterized its distinct antifungal mechanisms in Candida albicans, with an ultimate goal to combat opportunistic pathogens. And his research interest lies primarily in the areas of functional genomics, personalized medicine, development of evolutionary, genetic, and infectious disease. And when I read Sir's thing, there were so many words called novel. Maybe we should, when we refer the dictionary for Sir's name, we'll have a word called novelty. So we are very happy that you have joined with us today, sir. Over to you for the session, sir.
So can you able to see my screen now?
Yes, sir, your slides are visible, sir.
Sure, yeah. So thanks, Uma. Thank you so much for the nice introduction. And especially, unfortunately, whatever work I did, it's all novel. It's not specific that I'm more interested towards a novel work. So thanks for the nice introduction. So as Uma said, like I've been working as a research associate at Department of Human Evolutionary Biology at Harvard University. So I spent almost 4 years at Harvard University. So today I'm going to present one of the research findings that I mentioned in my biography, that I identified as 2 separate DNA mutations in the human genome that causes 2 different diseases. So before getting into my actual research findings, I just want to first of all thank all the organizing committee, and also I would like to thank Dr. Vaidehi Vijay Kumar ma'am, the Honorable Vice Chancellor, and also Dr. C. Hilda Devi, Registrar, and Dr. A. Usha Rajanandini, and Dr. D. Ilakyam ma'am. So I would like to thank all the organizer first of all, and first of all I would like to thankful for offering this opportunity, and I'm very grateful to be here. And I would like to especially thank Dr. D. Ilakyam ma'am, because she was the one who contacted me for this regarding this conference. And I also worked with Ilakyam ma'am. We worked in the same lab like for 5 years. I did my PhD, and Ilakyam ma'am was also there. So I know him very well, and this is a great opportunity for me. And also I would like to thank other faculties, students, speakers, and the participants of the Mother Teresa Women's University. So with this, I just want to give a brief introduction before moving on to my actual research findings. So as you all know that the human body, this is just the human body consists of 37.2 trillion cells. So it means like we have a number of cells, like, and all the cells, if you look at all the cells, they all are not identical. So they have the unique features. Like if you take the neurons, and if you take the cells from the heart, or if you take the cells from intestine or something, so it's all cells. But if you look at their features, they are completely different. Not all cells are same. And also the cells contain the hereditary material called DNA. So all the cells contain DNA, but if you look at the features, they are completely different. So what makes the difference? So the DNA, which is present in the cell, so this has the active and the functional and the non-functional regions in the genome. So this functional and the non-functional regions that varies among different cell types. So some of the regions in the neurons that are active, which is not active in the heart cells, and that may not be active in other cell types. So that define the specific cell types. So basically each cell, it contains hereditary material. And as you know that the DNA, it's compactly packed within the nucleus. It's in the form of chromosomes. And if you take the DNA from any of the cells, like you take DNA from any species, and if you just sequenced using a sequencer, the sequencer is an instrument, like you might be aware of the term next generation sequencer or the Sanger sequencing. So if you sequence the DNA within the cells, you would expect to see alphabets like this. So the DNA, it contains nucleotide bases such as adenine, guanine, cytosine, and thymine. So ATGC, so you can see this is how the DNA, the letters are arranged. So they are arranged in a precise manner. So if you look at these, for this I just made for the examples, but the DNA is arranged in a precise manner. And the precise manner in the sense like it should be in a particular order. If there is any changes in the sequence, like if any of the nucleotide bases get changes, that leads to severe biological effects. Something like it may lead to some severe disease phenotype, or it may be lethal to the organisms. So the arrangement of all the nucleotides in the DNA is very important. So if you look at these sequences, you can see this is the ATG, as you know about this ATG start, and this is the stop codon. So mostly the scientists around the world, they care about the genes. So if they sequence the DNA, they want to know where the genes are. So the genes have basic rules, like it has a start and stop codon. And also the genes have the triplet nucleotides that codes for amino acids. So all the coding sequences that starts with ATG and the TGA, the start and stop codon, that codes for a protein. So most scientists, they care about genes. The reason is like the genes can be easily studied. The reason, the genes are functional. So the genes codes for protein, it encodes for RNA. So it's all these coding sequences are functional. So these can be easily studied by using the RNA sequencing technology or exome sequencing technology. But the problem here is, if you look at the human genome, 98% of the human genome are non-coding sequences, only the 2% that contributes for the coding sequences. So it means we are just focusing only on the 2% of the human genome, and we ignore about the 98% of the sequences in the genome. And like if you look at the data, like the latest technologies, the genome-wide association studies, so all these genome-wide association findings, what it says, it's like nearly 90% of the disease are associated with some of the genetic changes associated with the non-coding sequences. So we just only care about the coding sequences and we ignore all the non-coding sequences, which is really important to understand the disease mechanisms. As I mentioned, like the coding sequences have the triplet nucleotides. The triplet nucleotides I just mentioned in this table. So all these triplet nucleotides that encodes for amino acids. So that in turn produces a protein. So all coding sequences expressed and it produces a protein. But that is not true for non-coding sequences. The non-coding sequences doesn't follow any of the basic rules, such as the non-coding sequences, they don't have any triplet nucleotide that doesn't code for any of the proteins. So there is a difficulty in understanding the genetic changes associated within the non-coding sequences. So you may ask a question like why it's important to study the non-coding regulatory sequences. As I mentioned, only 2% of the human genome contains the coding sequences and remaining 98% of the non-coding sequences. So the non-coding sequences are, it's important to investigate the reason. The non-coding regions contain regulatory sequences. We call as regulatory switches. It's something like a switch I showed here. So these regulatory switches are present within the non-coding sequences. What happens? These are the switches that controls the gene expression. So this acts something like a switch. If you turn on the switch, that on the gene expression. If you turn off, that offs the gene expression. So all these regulatory switches are present in the non-coding sequences that controls the gene expression. So if something went wrong with this regulatory switches, for instance, if some mutation or some genetic changes happen within this regulatory switches, what happens that affects the gene expression? So if this differences in gene expression, that leads to human variation. So this is the reason why not all humans look similar, because we all have the cells, we all have DNA, but we all, like except the twins, like if you look at the human population, they don't look similar, because this is because of difference in gene expression. And also this is the difference in gene expression also responsible for the morphological differences. That's why humans are different from chimpanzee and we are different from other species. And also this difference in gene expression that leads to human diseases. As I mentioned, these switches are very important, the regulatory switches in the non-coding regions. If something goes wrong with this regulatory switches, for instance, wrong in the sense some mutation or something happened with this regulatory switches in the genome that affects the gene expression that leads to a phenotype such as like human variation that can cause morphological differences. And also like it leads to human diseases. So here I'm showing one example how the switch operates. The regulatory switches are nothing. They are just the DNA sequences, just a short DNA sequences that activates the gene. So if you look at this image, so I mentioned here, this is the gene X. So the black bar here is the gene X. Let's take this is the gene. And you can see the switches are here. So these are different switches. They are not the same switches. So one switch here, the triangle, the red highlighted triangle. So this controls the gene expression in the hind limb. So if this switch is on, and that makes this gene to express in the limb buds, and there is another switch nearby. If this switch is on, that activates the gene expression in the neural tube. And also like if you can see the another switch here, if this switch is active, that activates the gene expression in the brain. So you can see like there are different switches, but it controls the gene expression in the different regions. But this is the final product of all these expression. So this is just a mouse embryo I'm showing here. If you see here, if you just blindly see this mouse embryo, you can see the expression of the gene. So all the blue highlighted regions are genes. But this is not because of one switch. You can see multiple switch operate at a different location. For instance, the limb bud and also the neural tube and also the brain. So the product of all these switches is you are seeing at the final image. So what I'm coming to say here, there are different switches that activates the gene expression in different parts of the body. So even though it's the same gene, but the switches that operates the gene in the different regions of the body. So this is just a basic information I'm providing that will help you in understanding the actual research. So with this basic information, as I mentioned, my current research project is focused on the two of the most common musculoskeletal disease in the world. One is the knee osteoarthritis. I think you might be heard of this term, knee osteoarthritis. It is a joint pain, and this happens in elderly people. And also I focused on other musculoskeletal disease called a hip dysplasia, which happens in the newborn babies. So my main question is to understand the genetic changes or the genetic mechanisms that contributes to knee osteoarthritis and hip dysplasia. This is my main question. So this is a little bit about the hip dysplasia and knee osteoarthritis. As I mentioned, I currently focused on two of the most common musculoskeletal disease in the world. One is the hip dysplasia, which is called as DDH. DDH is the developmental dysplasia of hip. And another disease I'm focusing is the knee osteoarthritis. So if you look at the humans, so this is how the normal humans looks like. This is the hip. I'm just showing the hip here. So you can see this is mentioned as the hip socket, and this is the bone called femur, and the femur has a ball-like structure. So in the normal hip, if you take a normal individual, the hip looks like this. So what happens, the femoral head that sits and freely rotates within the hip socket. So this is a normal hip. But in case of hip dysplasia, what is happening here is you can see the femoral head, it dislocates from the hip socket. So this condition is called a DDH, and the DDH is also known as unstable hip. And this DDH, if you look at the heritability, nearly 55% of it, this disease is approximately 55% of heritable. So this has more heritability compared to the knee osteoarthritis. And also, if you look at the frequency, like 1 in 1,000 newborn babies have this hip dysplasia. So this is just, if you just look at 1,000 babies, at least one baby might have developmental dysplasia of hip. And now you might be clear with what is hip dysplasia. This is a dislocated hip, or it's an unstable hip. And if you look at the knee osteoarthritis, so this is how the normal human knee it looks like. You can see, so this is a bone called, this is a distal femur, and this is a bone called distal tibia. This is a scientific term. So this is how the normal knee, you can see it looks intact. But in case of knee osteoarthritis, what is happening here is, this is a degenerative disease. You can see the degradation of cartilage. You can see all the cells are degraded in the knee. So this disease condition is called knee osteoarthritis. As I mentioned, the joint pain happens usually in the elderly people. So nearly 30% of the population over 65 years, they might have the problem of getting knee osteoarthritis. And also the heritability is approximately 40% of this knee osteoarthritis heritable. So this is about the two diseases I'm currently working on. And you can see a colorful plot here. So I'll explain what this plot is. Initially in my presentation, I was talking about the genome-wide association study. So the genome-wide association study, I'll quickly explain what it is. So you have a healthy individual and you have a diseased individual, we call as a patient. So you are sequencing the genome of the normal healthy individual, and also you are sequencing the genome of the diseased patients, like the patients. And if you compare the genomes, both are humans, but one is normal, one is patients. And if you compare the genome after sequencing, so you likely do see some of the changes in their genome. For example, you have like a normal genome, and also you have a genome from patients. If you compare them, you would expect to see a difference. Consistently, you will see some difference. So that difference is likely the reason for the particular diseases with respect to the genome-wide association studies. So if you look at this plot, what I'm showing here is, so these are all the chromosomes, like 1 to 24, I mentioned, this is all the chromosomes. And this is all the p-value, that is, we do some kind of statistics. And all the dashed line here, this is a threshold. We call as a genome-wide significant threshold. So what you can do, you can sequence the genome from the healthy individuals, and also you can sequence from the patients. And if you compare them, and if you plot in a graph, you can see a dots here. So all these dots indicates the genetic changes associated with this region. If you just take this region, it's a chromosome 1. And all these dots here, it indicates the genetic changes that is present in chromosome 1. Similarly, it's chromosome 2. So if you look at these data, so you can see some of the green dots here. These green dots has crossed this dash like a threshold. It means this is a significant threshold. Whatever the dots which is above this threshold, we consider this as statistically significant. So we can it means like it is worth investigating those changes because this is might be likely associated with the disease. So basically, these two plots, it shows the genome-wide association studies which is performed on hip dysplasia and also the knee osteoarthritis. And you can see here some dots here, and also you can see some of the dots. So you can see some of the dots here. So these are some of the genetic variants we called as, you can call a single nucleotide polymorphism or the genetic changes, or you can call this as like a DNA mutation. So these are the mutation which falls within one gene called GDF5. GDF5 is a gene called a growth differentiation factor 5. So basically, these data comes from like a millions of individuals. It's not from one patient. So you have to sequence like a healthy individuals, and also you have to sequence a patient. So you have to, it's like a population level. It's not like from one patient. So in both cases, what we identified is like, you can see some of the mutations here and also some of the genetic mutations here that falls within the one gene called GDF5, which is a growth differentiation factor 5. This has been associated with hip dysplasia and also the knee osteoarthritis. So we know these regions are associated with the disease phenotype. So the next thing we want to know, so you can ask the question like why the GDF5 gene is important, because you can also see the other dots here, but this dots here, these are the genetic mutation that has reached the significant threshold. That's why we want to focus on this particular gene. And if you look at the GDF5, as I mentioned, GDF5 is one of a protein that is really important for the skeletal development. And this protein is a bone morphogenetic protein. As I mentioned, it plays an important role in skeletal development. If you see, the GDF5 protein is expressed. Expressed means this is present in the hip. For example, here I'm showing the image of the hip. The GDF5 protein is expressed in the hip. So this is one bone called femoral head. As I mentioned before, it looks like a ball-like structure, and this is the socket. So the ball, it sits and freely rotates within the socket. So the blue color here, it indicates where the GDF5 protein is expressed. So basically what you are seeing, okay, you are seeing the GDF5 expression in the hip, in the proximal femur, and also in the acetabulum. And the next thing what we did is like, we just knock out the GDF5, knock out in the sense you delete or you remove the GDF5 gene from the mouse. So what you observe here is this is the normal mouse. Normal mouse in the sense the mice is having the normal GDF5 gene. So we remove the GDF5 gene from the mice, and what we observed here is if you remove the GDF5 gene, you can see if you compare this and this, the ball-like structure, the size it decreased. So it has a shorter femoral head, and also if you compare these two, it becomes a shallow acetabulum. So this GDF5 has a really an important role in hip joint formation. Based on this data, you can see if you remove the GDF5 gene from mice, that shows the hip defects. And also the previously lot of researchers have done research on the hip dysplasia. So the image I'm showing here is the X-ray image from the human patients. So the human patients, they sequenced the gene and they found that the GDF5 gene is having mutation. So this clearly shows the GDF5 mutation in human that causes the hip dysplasia. So this is same with the knee osteoarthritis, the other disease. For example, you can see the GDF5 gene is expressed in the knee surface. So this is the knee which forms the femur and tibia. These two bones form the human knee. So you can see the GDF5 expression in the knee surface. And also if you delete the GDF5 gene in mice, for instance, if you remove the GDF5 gene from mice, that leads to knee defects. And also the human patients which is having the mutation in the GDF5 gene also show the knee osteoarthritis phenotype. So this clearly shows the GDF5 protein is important to investigate with respect to the hip dysplasia and also the knee osteoarthritis. So we know the gene play an important role, but our goal is to understand the genetic changes that is associated with these two diseases. So what I did here is, this is a bit complicated to explain. So I'll just explain. This is a view called UCSC Genome Browser view. So here you are looking at one regions of the human genome. For instance, let's take this is a human genome. In the human genome, you are just looking at the one portion of the genome, not the complete genome. So if you look at these, this is the GDF5 gene in the human genome where it is located. And this is the next gene. This is the neighboring gene which is called as UPCC. So what basically we did here is we took the human genome and we just focused on the GDF5 gene, which is here. And then, as I mentioned, the genome-wide association study has been performed on the knee osteoarthritis and the hip dysplasia patients. So we took all the mutation and the genetic variants associated with the knee osteoarthritis and the hip dysplasia. Basically, the line you are seeing, the dark line, the black line you are seeing here is a genetic mutation. Let's assume this is one genetic mutation. This is another DNA mutation. Likewise, you can see all these are the DNA mutation that falls within this region in the human genome, the GDF5 gene and also the UTCC. So all these are the mutation associated with the knee osteoarthritis. And also these two, you can see it's highlighted in a different color. These are the genetic mutation associated with the hip dysplasia. So basically what I did here is we took the human genome, we just focused on the GDF5 gene, and we mapped all the genetic mutation to this region. So now you can see there are a number of DNA mutations. Hundreds of DNA mutations falls within this region. So the real problem here is we don't know which is the real mutation that causes disease, because this is not just one mutation. There are hundreds of mutations that has been associated with knee osteoarthritis and also hundreds of mutations for hip dysplasia. We don't know which is the causal mutation, which really causes the disease phenotype. So this is our main question. So we want to know what is the molecular basis of the observed disease phenotype. So now you might understand the question, because here we have so many DNA mutations near the GDF5 gene. We want to pinpoint which mutation actually caused the disease. So that's our question. So in order to answer this question, we use a technique. So this technique is a popular technique nowadays. Every lab has been doing this. This is called as an attack sequencing. So the attack sequencing is a next generation sequencing technique. So using this next generation sequencing technique, you can profile the open chromatin regions in the genome. Open chromatin regions in the sense, I'll explain this figure. Let's take this as a human genome. So if you look at the human genome, some of the human genome are, it's called as closed chromatin. So this region is closed and some of the regions are open. So this region is open, we call as open chromatin. This is called a closed chromatin. So if you use this attack sequencing, what exactly this technique does here? This technique makes use of a special enzyme called TN5 transposase. So this is just basically, it cuts the DNA. So this TN5 transposase that cuts the open chromatin regions. It cannot cut the closed chromatin because this region is closed. So it cuts only the open chromatin regions. You can see it's cutting here. Once it cuts the open chromatin region, it adds a small piece of a sequencing adapter to this region. So you can see here, the enzyme cuts here, and after cutting, it just added the sequencing primer. So using the sequencing primer, you can able to sequence specifically these open chromatin regions. Because closed chromatin regions are closed, it's compactly packed with the nucleosome, so these regions are not accessible. So basically, if you use this attack sequencing, you can able to sequence the open chromatin regions in the genome. I'm not talking about one specific region. You are sequencing all the open chromatin regions in the complete human genome. So now you can ask the question, why we need to sequence the open chromatin region? Because the open chromatin regions are the regions which are functionally active in the genome. For example, if you take a heart cells, if you take a neurons, and if you perform attack sequencing, so the region which are open in the neurons, it may not be open in heart cells. So the region which are open in the heart cells may not be open in the neurons. So that defines the cell types. So if you profile the open chromatin, so that says the region that are functionally active in that particular cell type. So I used an attack sequencing approach. So what I did here is, so you know the question before, we have a lot of genetic mutation, DNA mutations. We don't know which is the real mutation that causes disease. So I perform an attack sequencing on the hip. So here the image I'm showing is the skeleton of the embryo, human embryo. So we use a particular time point at E54 means embryonic stage 54 days. So after aborted, the hospital sent us the human embryo and we collected the hip region. So this is the hip. And we collected the knee region. So the hip contains, as I mentioned, one bone is the acetabulum, which is the hip socket, and this is the proximal femur. And the knee contains the two ends of the bone. One is the distal femur and another one is proximal tibia. So basically what we did here is we collected these samples like the acetabulum, proximal distal, proximal femur, which forms hip, and also the elements that forms the knee, and we perform this attack sequencing. So using attack sequencing, as I mentioned, you can profile the open chromatin regions or the regions which are functionally active in the genome. So once we perform the sequencing, next what I did here is, this I showed before. So I showed this is the UCSC genome browser view. You may be familiar with my previous slide. So this is the GDF5 gene, and this is the neighboring gene. So this is our human genome. You can see the GDF5 gene and UQCC. So I just zoom in this particular region in the human genome. These two already you saw. So these are the DNA mutation associated with knee osteoarthritis. And these two, the color which is highlighted in difference, are the DNA mutation associated with the hip dysplasia. So you can see the blue highlighted regions are our attack sequencing results. As I said, we perform attack sequencing on the hip and knee. So this is the attack sequencing results. So you can see there are black bars. So this, if you use an attack sequencing, if you analyze the data sets and if you open in this browser, you will see a black bars here. So the black bars, what it indicates here is, so these regions are functionally active in the genome. For example, if you take this bone acetabulum, so these regions like 1, 2, 3, 4, 5, 6. So all these regions are open in the genome. If you look at the other tissue like a proximal femur, you can see this region is functionally active. This region is functionally active, which is not active in the acetabulum. Basically, what we did here is we intersected all the results together. So this is the DNA mutations associated with two disease, and this is our attack-seq results. And you can see here this R1, R2, this GRO1. So these are the enhancers. As I mentioned, these are the regulatory switches. We know these regulatory switches that controls the gene expression. So basically, by intersecting all these results, what we identified here is, I highlighted two regions here with a red line. Here is one region and here is one region. So one of the DNA mutation here that falls within one of the regulatory switch, that is an enhancer called GRO1 enhancer. And also the other genetic mutation, DNA mutation here, that falls within another enhancer, that is a regulatory switch called R4 enhancer. And also, if you look at these regions are functionally active in the genome. So we want to know whether this region is active. You can ask the question, other mutations are here, why I didn't consider the other mutation? The reason is, if you look at these mutations, that is not active in the genome. So it's not worth investigating those DNA mutations. So these are the mutation that falls within a GRO1 enhancer, and also that region is functionally active, because we should care about the region which are functionally active in the genome. So basically, by intersecting all our data sets, what we identify is, like, we identify two regulatory switches. Regulatory switches in the sense it's an enhancer that controls the GDF5 expression. One enhancer name is called GRO1, another enhancer name is called the R4. So now we know the DNA region, and also we know the DNA mutations that falls within the GRO1 and R4 enhancer. Next, we want to know where this GRO1 enhancer is expressed. So what we did here is, we fused a reporter in the sense, we want to know where this GRO1 enhancer is expressed in the human and the mouse embryo. So here you are seeing an image for the mouse embryo, and this GRO1 enhancer is expressed strongly in the hip. You can see the blue color highlighted. This is the bone that forms the hip. This is the femur and hip socket. Basically, this GRO1 enhancer is active in the femoral head and also in the acetabulum that forms the hip. And also you can see another enhancer here, R4 enhancer. This R4 enhancer is expressed in the knee joint. So this is the region which is highlighted in blue. So that's the knee joint. Basically, if you look at the GRO1 enhancer, the GRO1 enhancer is strongly expressed in the sense it's more active in the hip, but it is not active in the knee. And the another enhancer, it's a regulatory switch called R4, that is expressed or active in the knee, but that is not active in the hip. So you can see that GRO1 is specific for the hip, the human hip, and the R4 enhancer is more specific for the human knee. So this is the mutation I said, the mutation that falls within the GRO1 enhancer. So usually, normally, if you look at the humans, I'll show these data sets. So this is the mutation that falls within the GRO1 enhancer. So basically what we did here is, we took the DNA sequence from different species. You can see it's a human, chimpanzee, it's gorilla. So we took the DNA sequences from different species and we did the multiple sequence alignment. So basically what you are seeing here is, you can see this is the G nucleotide. The G nucleotide is highly conserved. All the species have G nucleotide in this position, including human. But interestingly, some of the humans have the A mutation. So we don't know why those humans have the A mutation. So, but naturally, if you look at the human, so the G nucleotide should be in that position. But some humans, interestingly, they have the A mutation. And this A mutation, it occurs in high frequency in European and Asian population. So this is for the mutation that falls within the GRO1 enhancer, which is associated with HIP. So the next mutation I was talking about is within the another enhancer called R4 enhancer. So again, what we did is like we took the DNA sequence from different species, we performed a multiple sequence alignment within this R4 enhancer. So what you are seeing here is the C nucleotide is highly conserved in primates. If you see human, chimp, gorilla, C is highly conserved. But interestingly, some of the humans have these T mutations. We don't know what's the reason why these humans have T mutations. But naturally, human should have the C nucleotide at this position, but some humans have the T mutation. So we want to investigate more on this mutation. So hereafter in my rest of the presentation, if I'm talking about the risk variant means G, which is the non-risk variant means G, it means that is naturally present in human. The risk variant means A, but that causes a disease phenotype. Similarly, like the non-risk variant in the R4 enhancer is C and the risk variant is T. So just to differentiate between the mutation and the non-mutation. So now basically what we have here is we have a two enhancer. One is GRO1 enhancer and another one is the R4 enhancer. And we know that mutations that falls within the two enhancer. Now we want to know, as I mentioned, like my field of expertise is on functional genomics. We want to functionally test what is the function of these region, like the GRO1, R4, and also what's the role of this mutation in the human genome. So my first question here is, as I mentioned, I want to know what is the function of this GRO1 enhancer in the human hip joint formation and the hip dysplasia diseases. And also I want to know what is the role of R4 enhancer in the knee joint formation and the osteoarthritis risk. So in the previous presentation, you have heard about the term gene editing using CRISPR. So this shows like we have used CRISPR a lot in our research because this is a functional genomic approach one can use to investigate the function of the regions in the genome. Like it can be used for any species. In the previous presentation, the Professor Reddy sir have shown how you can use CRISPR in the plant system. Here we use the CRISPR in the mice model, and also we use the CRISPR in different cell lines so that we can better understand the function of the region. Like I'll explain in the next stage. So this is how the CRISPR works. You have your target DNA, and here you are using a guide RNA. The guide RNA is the RNA where you are, suppose you have a target DNA, you want to delete this particular region. So you have to design a guide RNA, the region which is complementary to the region that you are going to delete. So this is a guide RNA which guides an enzyme called Cas9. Cas9 is an endonuclease that will cut the DNA. This is just like a scissor. So what happens, this guide RNA, you know where you want to cut your DNA or edit the DNA. You have to design the guide RNA based on the region that you want to edit. And this guide RNA will guide this enzyme to the region, like where it has to make a cut. Basically, the guide RNA takes the Cas9 to this region, and the Cas9 enzyme, it cuts the DNA. So this is how the CRISPR gene editing works. So to answer my first question, what I did here is, so as I said, I want to know what is the function of this GRO1 enhancer. This is one of the regulatory switch. So we used a CRISPR-Cas9 technology and we delete the GRO1 enhancer region in the genome. Delete in the sense you are taking out this DNA sequences from the genome of the mice. So once you remove, we call this as a GRO1 knockout mice. GRO1 knockout in the sense this mice is lacking this GRO1 enhancer. So now we have the mice which is not having this enhancer. Then what we did here is we want to know whether this GRO1 enhancer, if you remove the GRO1 enhancer, that is affecting the GDF5 gene expression or not. So we did a functional assays, and what we found here is if you delete the GRO1 enhancer or the GRO1 switch regulatory switch in the mice genome, that decreases the GDFI expression by approximately 19% in one of the bone end called proximal femur. This forms the hip. As I mentioned, this GRO1 enhancer is specifically expressed in hip. So if you delete this GRO1 enhancer in the mouse, that affects the GDFI gene expression. And also, as I said, we have this mice. So one thing what you can do is, because we do a lot of bone work, so the bone is like in humans, you know, right? You can just scan the bones, you can do the X-ray. So similar thing you can do. You can also scan the hip and the knee of the mice and see if you delete this GRO1 enhancer, whether that is causing any shape defects, whether that is affecting the hip or knee. So here, the data I'm showing here is basically the quantification data from micro-CT. The micro-CT is the scanning method we use to scan the bone. So we have the knockout mice, which is not having the GRO1 enhancer, and we quantified some of the features in the hip. So what we found is, like, if you delete this GRO1 enhancer in the mice genome, you can see that affects the femoral head diameter. And this is one of the features which is highlighted here. So we just did quantification here. So if you delete this enhancer, that affects these two regions. This is significantly affected. And these regions were not affected. So you can clearly see if you delete this region, GRO1 region from mice, that affects femoral head and femoral neck length. This is the region where the enhancer is expressed. The blue color is the region where this GRO1 enhancer is expressed. Basically, if you delete it, that affects this region. And this region, you can see, we use this as a negative control. So if you delete this GRO1 enhancer, so that's not affecting this region because this region is not expressing the enhancer. So what we want to convey from this slide is this have a specific effect. If you delete this GRO1 enhancer using CRISPR-Cas9 technology in mice, that affects only the region where the enhancer is expressed, that not affecting the region where no enhancer expression. So this reveals a modular enhancer function. So it's more specific. So this is for the GRO1 enhancer. So we did the same thing for the other enhancer, R4. Similarly, we used a CRISPR-Cas9 technology and we delete the R4 enhancer. Basically, we remove this enhancer from the mice. So the mice is not having the R4 enhancer. And again, we look at the GDFI expression. So this is a gene and this is a regulatory switch that turns on the gene. If you delete this R4 enhancer in mice, you do see decrease in GDFI expression because R4 enhancer is controlling this GDFI expression. So if you remove this switch, that affects the GDFI expression. And also we perform the micro-CT scanning. As I said, this is a mice which is not having this R4 enhancer region. We want to see if you delete this region that is causing any defects in the mice knee. So we quantified some of the features in the knee. So these are the significant changes. Like all these dots indicates like we did a quantification using the micro-CT. And these are the different genotypes of the mice. Basically, what we saw here is you can see a significant changes in condylar curvature and the notch width. So this is the region. This is basically the knee I'm showing here. So this region get affected, these two, if you delete this R4 enhancer. And this region was not get affected because there is no enhancer expression. So this is basically for the knee. And this R4 enhancer I mentioned, this is specifically expressed in knee and not in the hip. So we also quantified the hip and see if you delete this regulatory switch that's affecting the hip, but we didn't see any of the hip defects. So basically from this two slide, what you can learn, GRO1 is specifically controlling the hip joint formation and the R4 enhancer is specifically controlling the knee joint formation. So if you delete this from the human or mice genome, that specifically affects the hip and knee. So whatever I explained so far, it's on mice. You can see I use a CRISPR-Cas9 technology on mice. So the mice is not a human. So we want to know the same thing is happening within the human system. So the second question here is we want to know whether this GRO1 and R4 enhancer controls the GDFI gene expression in human chondrocytes. Basically, we cannot use the CRISPR-Cas9 on humans because it's not ethical. So what we did here is we used an in vitro system. So we grown the human chondrocytes. Chondrocytes are the bone cells. So we grown the chondrocytes in the dishes and we perform the CRISPR-Cas9 gene editing on these chondrocytes. Basically, I did the same thing here. So previously, whatever I showed is all on mice. So we use a CRISPR-Cas9 technology and we deleted the GRO1 enhancer, R4 enhancer in the cells, that is the human chondrocytes. They all are bone cells. So we use a CRISPR-Cas9. You can see I deleted the GRO1 enhancer, I deleted the R4 enhancer, and also the CRISPR-Cas9 technique is more precise. You can even delete like a few base pair. So this is the mutation region. As I mentioned, the GRO1 enhancer is having a mutation in humans. So this is the region that has a mutation. So we used a CRISPR-Cas9 and delete precisely the mutated region. And also we use CRISPR-Cas9 to delete the mutated region within the R4 enhancer. Basically what we did here is this is GRO1 and this is the mutated region we deleted. So we did a CRISPR with different combination. So in all cases, what you are observing here is if you delete the GRO1 enhancer, or if you delete the R4 enhancer, or if you delete the mutated region within GRO1 enhancer or the mutated region within R4 enhancer, in all the cases, you can see the GDF5 gene is going down. So if you remove the switch, basically you can call this as a GRO1 switch, R4 switch that controls GDF5 gene. If you remove these switches, what happens? The switch is not there, so the GDF5 expression is going down. So in all the cases, you can see the GDF5 gene expression is going down. Basically, what you can infer from this result is GRO1 R4 enhancer and also the variants that falls within these enhancers control the GDF5 expression in human chondrocytes. So this is what we learned from the previous results. The same thing we did in mice. We deleted all this enhancer and we found that mice is, if you delete that enhancer, that affects the GDF5 expression. That is same with humans. In humans, if you delete these enhancers, that affecting the GDF5 expression. So next thing we want to know what is the function of this variant, because as I was keep on talking about the mutation, we want to know what is the effect of this mutation. Let's assume this is GRO1. If some mutation happens within GRO1, we want to know what's the effect of that mutation on this GRO1 and R4 enhancer. So this is another technique you can use in lab. Basically what you can do, so we generated a 2 version of GRO1 enhancer. So one version is having the non-risk G variant and another GRO1 enhancer have the risk A variant. So basically, as I said, all humans have G, but some humans have A. So you can easily test the effect of this mutation in just the cell lines. You can grow the cells in lab and you can do the simple assay to study the effect of this mutation. So basically, we made synthesized a 2 version of GRO1 enhancer, one with the non-risk G and one with risk A. Similarly, we made a 2 version of enhancer R4 with the non-risk C and the risk T. The non-risk means it's naturally occurring in human, and the risk A and T are the mutation that happens in human. So basically, we made a different version of this enhancer. So this is just a plasmid vector. So as you may be familiar with the plasmid vector, so this plasmid vector, what happened? It has a gene called luciferase. This is a reporter gene. So let's take a first combination. You take a GRO1 enhancer with this G nucleotide and you clone it here and you transfect it into cell line and you measure the luciferase activity. So if this region is active, the cloned region is active, that activates this reporter gene luciferase that you can quantify using some of the assays. Similarly, let's take the second one. You made a GRO1 with the risk A variant and you cloned into this vector, and again you are measuring the luciferase activity. Similarly, you made 2 versions of R4 enhancer with non-risk C and risk T. You cloned here, and you are measuring the luciferase activity. Once you measure the luciferase activity, and if you plot it in a graph, you can see like the G variant, the GRO1 enhancer with the non-risk G, and this is a GRO1 enhancer with the risk A. So if the GRO1 enhancer have the risk A mutation, that is decreasing the luciferase activity, luciferase reporter. Basically, what it tells us here is in both cases, you can see the risk A variant and also the risk T variant that decreases the reporter activity compared to the non-risk variants. Basically, what you can infer from this result is if something goes wrong with these regulatory switches, for instance, all humans have the G mutation. G nucleotide in the GRO1 enhancer. So some of the humans have the A mutation. So if this GRO1 switch get mutated, what is happening here is this is an enhancer because it has some function in controlling the gene expression. If this regulatory switch get mutated, that is decreasing the enhancer activity that you are indirectly measuring by measuring the luciferase activity. So this is one of the functional genomic technique that you can use to study the activity of the fragments. Like you can study the effect of the mutation on the gene regulation. So the next thing, this is a more complicated thing we did here. So I hope the previous presentation would be very helpful. The CRISPR-Cas9 can be applied for any of the model systems. So here, what I said, like this is just a simple system. You can just make, you can use the recombinant DNA technology. You can do some cloning and test the effect of this mutation in just the cell lines. So what we did in our lab is we created a mice model. So we created a 2 mice model. Basically what we did here is we created the human mutation in mice. So this is just the 1 base pair mutation. This is not easy to create because the CRISPR-Cas9, still this technique is like, as it's a more popular technique. And we use the CRISPR-Cas9 and we precisely created a 1 base pair mutation in mice. Let's take the mice is having a genome. And in the complete genome, we just mutated the G nucleotide to A. So this is the human mutation. Similarly, for the R4 enhancer, what we did is the mice have the A nucleotide. So we created a A to T mutation. Basically, we used CRISPR-Cas9 and created the human mutation in mice. So this is the nucleotide which is naturally present in mice in GRO1 and R4. We use CRISPR-Cas9 and we created G to A mutation and A to T mutation. So these are the mutation that occurs in human. So we cannot create this mutation in human. So we used a model system mice to create this mutation. So now what we did here, we created the mutation in mice. And once we created the mutation, we quantified the GDF5 expression because as I mentioned, this GRO1 and R4 enhancer controls the GDF5 gene expression. If you create a mutation, we want to know this mutation is affecting the GDF5 expression. That's what I'm showing here. So if you create a G to A mutation, that is affecting the GDF5 gene expression by approximately 14%. This is specifically in the hip because this GRO1 is expressed in hip. Similarly, if you create the A to T mutation, that affects the GDF5 expression. So it decreases GDF5 expression by approximately 16%. Basically, what is happening here, if you're creating the mutation that is affecting the gene expression, GDF5 gene expression, if the GDF5 gene expression is get affected, that is causing like the disease phenotype. And also, as I said, we perform the micro CT scanning. Once you have this mutated mice, you can take the bone from the mice and you can scan the bones and you can see the bone having any defects or not. Basically, what here you are seeing here, the circle region is here, is the region which is affected because of this mutation. So if you create a G to A mutation in the GRO1 enhancer, that is affecting the region. So the region which is highlighted in red are the regions which are most severely affected. So this affects the proximal femur and the acetabulum that forms the hip. And the R4 mutation, if you create the A to T mutation in R4 enhancer, that is specifically affecting the knee. So this region is highlighted in the red. So this basically shows the region which are strongly affected, the red. So this clearly shows if you create the human mutation is in mice. So all these mice have defect in the GDF5 gene expression and also they show the disease phenotype, the hip dysplasia and the knee osteoarthritis. So this is with respect to mutation. So from my whole story, one thing is very clear, like the GRO1 and R4 enhancer is specifically expressed in the relevant tissue. For example, GRO1 is in hip and the R4 enhancer is in knee. And any of the mutation in the regulatory switches that affects the GDF5 expression and that causes specific disease phenotype. So now you may ask the question, just simple mutation, how this causes the disease phenotype? Because we didn't provide any of the mechanism or any pathogenesis mechanism to show that this causes the disease phenotype. So the last thing I want to just show here is how we define the mechanism for this, because this is just a mutation. So if this mutation happens within the body, we have to know how this really causes disease phenotype. So in order to go further, what we did here is, as I mentioned, these are the regulatory switches that regulates the gene expression. we want to know any of the transcription factor. The transcription factors are nothing but a protein that binds with the DNA and that regulates the gene expression. So basically, we want to see any of the transcription factor bind at this position or any of the transcription factor bind at this position. Because if the transcription factor bind at this position so that this mutation can disrupt the transcription factor binding, so that may cause decrease in GDF5 gene expression. So in order to investigate further, what we did here is we took the DNA sequence around this region, and also we took the DNA sequence around this region, and we did a computational method, computational prediction, to see any of the transcription factor, that is a protein that activates gene transcription. We want to see is there any transcription factor bind at this position. So interestingly, our analysis found one particular transcription factor called PIDX1. PIDX1, it's a master regulator of the hind limb development. So this is what is known about the transcription factor. You can see the PIDX1 transcription factor is expressed in hip. So this is the region of hip, and also it's expressed in knee. And this is already shown that if you remove this PIDX1 transcription factor from mice, that shows the hind limb defects. So this is just the mouse skeleton. What you are seeing here is, if you completely remove the PIDX1 transcription factor from the mice genome, you are seeing the hind limb get affected, but the forelimb remains normal. So our method identified the PIDX1 transcription factor. And what we did is, like, we use, this is another technique you can use to study the DNA protein interaction. As I mentioned, my question is to know, is there any transcription factor bind at this region? We used a technique called CHIP. It's a chromatin immunoprecipitation. Basically, this technique would allow us to study the DNA protein interaction. Let's take this is the DNA and this is the protein. So in my case, let's take this is the GRO1, and I want to know whether the PIDX1 transcription factor bound to this region or not. Basically, this CHIP assay, how it works is, you will cross-link the DNA and protein within the cells. So basically, you need to take the cells from the tissue and you need to treat the cells with a formaldehyde that will cross-link your DNA and protein. Cross-link in the sense, this is get fixed. So wherever the protein binds to DNA, that still remains. It forms a complex. It cannot be disrupted. Then what you can do, you can use a protein-specific antibody. Like this is an antibody that binds to the specific protein. So if you pull down the protein, you would expect to see the DNA that is bound to the protein. So this is how we identified that the PIDX1 transcription factor, it bounds to the GRO1 and the R4 enhancer. So this is just the CHIP assay. We just want to explain, this is a pull down. Pull down in the sense, we use the PIDX1 specific antibody and we perform the CHIP assay. And what we found is the PCR band here, it indicates that the PIDX1 is bound to the GRO1. And also the PCR band here, it indicates that PIDX1 is bound to the R4 enhancer. So here what we did here is we used a mice. Let's take this GRO1. So this GRO1, we made a mice in such a way that GRO1 region is having both the G and the A nucleotide. So this PCR band would contain both A and G nucleotide. So what we did here is we took this PCR band and we sequenced it and we count how many GRO1 sequence have the G nucleotide and how many GRO1 has the A nucleotide. Basically, if you do this assay, you can tell this, we know this PIDX1 is bound to this region, but we want to know, is there any differential binding of this PIDX1 to this nucleotide? Basically, what we want to know, let's assume the PIDX1 transcription factor bind in this region. Suppose the human have this A mutation. We want to know if this mutation that affects the binding of the PIDX1 transcription factor. That is our goal. And using this CHIP assay combined with next generation sequencing, we found that the mutation, the A and the T mutation in these two enhancers, that affects the affinity of the PIDX1 binding. So whatever I showed, this is my last results I'm just going to show. Whatever I showed so far, it's all like we performed several experiments on mice and we perform on cell lines. Finally, whatever we do, we want to, because it should be translated, it should have real application in humans. So we want to confirm all our findings in the human patients. So this is my last data I'm showing here. So what we did here is we recruited DDH patients, like 113 DDH patients. DDH patients, they have hip dysplasia, you can see. So we collaborated with hospital and we recruited DDH patients. And also we recruited for patients who are having the osteoarthritis. So basically we have a patient from DDH and the osteoarthritis patients. We did two things on each patient. One is genotype. Genotype is we know that the GRO1 switch is having mutation. As I mentioned before, G to A mutation, A is the risk mutation that causes hip dysplasia based on the results I showed so far. So we collected the DNA samples from the DDH patients. We genotype them. Genotype in the sense we sequence the region that contains the mutation in GRO1. And what we observed is the people with the DDH, the people who are DDH patients, they have more of A variant. A variant means they have the more of mutation, the A mutation in their genome. This confirms our previous results. As I mentioned, G is the normal nucleotide that present in all the humans. If it gets mutated, like if the G nucleotide becomes A mutation, that causes hip dysplasia. So this is one of our findings that confirms our all the results, whatever I showed before. All the DDH patients, they have like A mutations. And also you can see nearly, we sequenced 113 patients. Among 113 patients, 112 patients, they have A mutation. So this shows a strong evidence. If a human have the A mutation, they might get hip dysplasia. And also we have the X-ray images from all the patients, and we quantified one of the features called acetabular inclination. This is just like an X-ray. You can take the patient's X-ray and you can look at the acetabular inclination. So what you can see here is, this is the humans having A and G. This person is a heterozygous individual, and this is a homozygous patient. Homozygous patient in the sense the patients have 2 copies of the A mutation, but heterozygous individual have 1 copy of A mutation and one is the normal G. So if you look at this acetabular inclination angle, you can see a clear difference. You can see the additive effects, because here just 1 A, you have effects here. Here it's becoming AA. So you are adding 1 more A. So you do see clear significant changes in the acetabular inclination. So this is another evidence which clearly shows that the A mutation in the GRO1 enhancer is associated with hip dysplasia. We did the similar thing for the osteoarthritis patients. So we quantified, as I said, the R4 enhancer have the mutation T mutations. So we classified the patients into 3 categories. One is patients who are not having the knee osteoarthritis, and the patients who are progressing to knee osteoarthritis. It means they are in the mid stage. And the last classification we did here is the patients who are already having the knee osteoarthritis. If you look at the frequency of this T mutation in all these 3 classes, you can see the people with no knee osteoarthritis have less T mutation, and compared to people who are progressed to knee osteoarthritis and the people who already have knee osteoarthritis. So this clear trend shows that people who have knee osteoarthritis have higher frequency of the T mutation in the R4 enhancer. Similarly, we did some kind of phenotypic analysis. We want to see whether all these osteoarthritis patients have defects in their knee. So this shows the knee gets affected. So with all these results, so this is our current model. I think this would simplify all my findings. So we have one gene. The gene is called GDF5, which is the growth differentiation factor 5. So basically, this is a gene. It's a coding sequence. And we have 2 regulatory switches. So these 2 are enhancer. One enhancer is called as GRO1. Another enhancer is called as R4. So this is how the things are organized in our human genome. So this GRO1 enhancer controls the GDFI expression only in the hip. And this R4 enhancer, it controls the GDFI expression only in the knee. That's why it's mentioned hip and knee. So if you look at the knee, as I mentioned, the knee is associated, the knee have the R4 enhancer because it's expressed GDFI expression in knee. So the mutation in the R4 enhancer, let's take this is the R4 enhancer. The mutation, T mutation in the R4 enhancer that decreases the transcription factor binding that I showed before using the chip assay. So basically what happened, you have the R4 enhancer, which is active in knee. If any mutation happen in R4 enhancer that decreases the binding of transcription factor. So if this binding get disrupted, that affects the GDF5 gene expression that leads to knee osteoarthritis. And if you take the hip, as I mentioned, the GROW1 enhancer is active in hip, but not in knee. So this GROW1 enhancer is a regulatory switch that controls GDF5 expression. So if any mutation happen, like the A mutation that happens in the GROW1 that decreases the transcription factor binding only in the hip, that leads to decreased GDF expression in hip joints that leads to hip dysplasia. Basically, what we did here is we identified two separate mutations, one mutation in the GROW1 that causes hip dysplasia and another mutation in the R4 enhancer that causes knee osteoarthritis. Basically, we uncouple the two. Here you can see from the first introduction slide, I said, like we have two switches that controls the same gene expression, but in different tissue. If the switch get affected or it's get mutated, that affects the GDF expression and that affects only the tissue where these switches are regulated. So this is the whole story of my findings. So similar to this, so the genome-wide association study I explained in the first slide. So the genome-wide association studies have identified a large number of genetic loci associated with complex traits and disease. So the one thing I focus today, it's only on the human skeletal disease. If you look at the other disease like cancer, immune system disease, like a neurological disease, cardiovascular disease, and other metabolic diseases, there are a lot of genetic mutations has been reported before. Like all these color dots, these are all the chromosomes, like 1, 2. So it's a chromosome, and all these color dots here, it indicates the DNA mutations are the genetic changes associated with different diseases. So you can see there are a number of genetic mutations that has been already identified in relation to different diseases. But the problem here is we don't know which is the real mutation that causes all these diseases. So by making use of a functional genomics tool, so this is one thing you can consider if you are pursuing the research in future. So the genome-wide association studies have been performed on different diseases like cancer, neurological disease, cardiovascular disease. So first thing what you can do is like you can generate a functional genomic data. So you can use all these techniques, whatever I showed in my presentation, like a fine mapping, epigenetic, like you can do chip sequencing, you can do the attack sequencing, and you can do allele specific expression. The chromatin Hi-C is a 3D genome. Like our human genome, it's also folded in the 3D configuration. So by using this chromatin interaction technique, you can study the 3D genome. So basically, the GWAS is something like a pool. You can see like these are all genetic mutations. If you want to identify one specific mutation, the first thing is you need to generate functional genomic data. So this functional genomic data would allow us to prioritize the nucleotide variant or the genes that is associated with disease. So in our study, as I showed before, we saw hundreds of variants associated with knee osteoarthritis and hip dysplasia. So we used an attack sequencing approach and we pinpoint 2 variants. Likewise, if still you have hundreds and millions of variants, if you have the functional genomic data, you can prioritize the variant that can be further tested. So how you can test those variants? You can use a CRISPR technique. Like you can generate a knockout mice model, you can create a base editing. As I showed, we created a single base pair replacement mice, and you can do the knock-in of fluorescence, somatics. So all these techniques you can use to test the function of these variants. In addition to this, you can also do the omics profiling. Now it's becoming popular. Like you can look at the whole transcriptome, whole proteome, or whole metabolome. And also you can use the state-of-art technology. This is the stem cell technology. Like you can combine the iPSC technology with the CRISPR gene editing technology, and you can develop some of the disease model for drug screening and cell therapeutics. Basically, the purpose of this slide is if you use all these techniques, if you integrate all these techniques, you can successfully interrupt and you can prioritize the variant from the GWAS studies that could leads to novel biology and disease mechanism for its translation. So the main application of our work is here, what we did is like we identified the 2 separate mutations, right? One mutation in GROW1, another mutation in R4. So this mutation is associated with hip dysplasia, and this is with knee osteoarthritis. In future, you can use this for the personalized medicine. For example, if you want to know like whether in future you will get knee osteoarthritis or not, so what you can do, you can sequence this region in the genome, and you can see whether your cells contain the T mutations or not. For instance, like if you know you have the T mutation in your genome, so you can adjust your lifestyle modification, because knee osteoarthritis is not only caused only because of mutation, it also the other environmental factors also contributes to the osteoarthritis. So basically, this technique would allow us to predict the risk of getting the disease in future. So with that, I would like to thank you. Thanks my lab members. So this is a lab I'm currently working on. It's a Capillary lab in Department of Human Evolutionary Biology. And also all these are our collaborators who helped us in performing all the micro CT scanning and also the other steps which is related to the morphometric analysis. So with that, I would like to thank you all for your patience in hearing my presentation. And if you have any questions, I'm happy to answer.
Thank you so much, sir. Dear participants, if you have any queries, you can ask sir now. Any queries?
Yeah, if you have a question in future, maybe you can write me an email, or if you need any help with any of like, so I'll just show you email address. So this is my email address. You can write me an email. Feel free to reach out to me at any time. So I'm happy to take any question right now, or if you have any question, write me an email. Or if you need any help with any of the supervision, if you are interested in doing PhD or applying for any fellowship or something, so I'm happy to.
Thank you, sir. Thank you so much for telling that you would help us also. Thank you so much, sir. Your presentation was so crystal clear, sir. I think that's why the participants do not have any questions. And thank you so much for that informative presentation, sir. And we were absolutely amazed, like how you could easily explain something that was so complicated. There was so much ease with what you explained. It was so easy, sir. And we were able to clearly understand the roles of GDF5 and GRO1 and R4 and hip dysplasia and knee osteoarthritis. And it is purely our luck to have a resource person like you who has worked so much in a Nobel Prize winning technology like CRISPR technology. So thank you so much for joining us, sir, in spite of your busy schedule. And we also know that it's midnight for you also. Thank you so much for joining us, sir. And we would like to have an organizer for thanking you, sir.
Pushpanathan, thank you so much, Pushpanathan. You are doing wonderful work in sequencing. I think it is very much useful to our research scholars. They can use these techniques in their work also. Thank you so much, Pushpanathan.
Thank you so much, ma'am. Especially
I'll catch you later.
It's my pleasure to be here. Thank you, ma'am.
Thank you so much, sir.
Yeah, if you have any question, write me. Thank you.
Yes, sir. Thank you, sir. So we would like to move on with our next session. And for this session, we have with us Dr. Shankar Manoharan, sir. And sir is passionate about microbes, microbial communities and their workings. And sir is currently leading the microbial physiology group at the Department of Bioscience and Bioengineering at IIT Jodhpur. Sir also holds a master degree in genomics and a PhD in microbiology from Madurai Kamaraja University and postdoc from Kansas Medical Center in USA. Sir was awarded the DBT Wellcome Trust India Alliance Early Career Fellowship in 2017 to study the regulation of virulence in the hospital-associated pathogen Klebsiella pneumoniae. Sir's group is also studying the human gut microbiota in the context of health and diseases. Particularly, his group is interested in understanding how environmental cues modulate the gut microbiota structure and function, and how these alterations can impact the host physiology. So we are very happy to have you with us, sir. And now the session is yours, sir.
Thank you, Maheshwari. And I would, at the outset, I'd like to thank all the organizers of this conference, for inviting me to be a part of it, and for allowing me to share the work that we do with all of you today. Can you please confirm if I'm clearly audible and my slides are visible?
Yes, sir, your slides are visible, sir.
Thank you. So, good afternoon, everyone. I know this is the pre-lunch session. All of us are hungry, so I'll try and keep my talk as brief as possible so that we break for lunch on time. I am Shankar Manoharan, and currently I am an assistant professor at the Department of Bioscience and Bioengineering at IIT Jodhpur. And so basically, IIT Jodhpur is located in the frontier city of Jodhpur in Rajasthan on the western end of the country. So what you're looking at is the sprawling campus of about 800 acres that we have on the National Highway to Nagaur in Jodhpur. So let me begin with a brief introduction to our institute and the kind of things that we do before I move into our research work. So the Department of Bioscience and Bioengineering at IIT Jodhpur basically conducts research in a lot of areas, including molecular physiology, environmental biotechnology, biomaterials, biophysics, molecular microbiology, microbial genomics, drug design, chemical biology, computational biology, molecular motors, and neuroscience and neuroengineering. So we have a very diverse group of faculty members who work in all of these areas. And these research areas, the work that we do in these research areas is actually contributing to technology that is being developed in the form of biosensors, new drugs being developed, systems biology models being developed for complex diseases, brain science being pursued as a part of one of the thrust areas of our institute, which is artificial intelligence. We're doing a lot of work in bioimaging as well as in nanomedicine, theranostics, bioremediation, and bioenergy, and even developing new algorithms for analysis of sequence as well as image data. So I joined IIT Jodhpur in 2017. I've been here for about 3 years. My PhD was from Madurai Kamaraj University, after which I had a 3-year stint in the University of Kansas Medical Center. So my interest has always been in microbiology. I've always been fascinated by the microbes, which are not even visible, but are able to kind of cause infections and bring down human beings. So all my research so far, including my PhD up to what I'm doing now, is in microbiology, although my PhD was in molecular plant microbe interaction, much along the same lines as Dr. MS Reddy was talking before. In fact, I attended the very first Asian PGPR conference that he once organized in Hyderabad. So after that, I transitioned into infection biology, where I learned about the gram-positive system with Streptococcus mutans, and right now I'm pursuing gram-negative bacteria again at IIT Jodhpur. So before we move into the technical details, I just want to introduce the area that I'm going to be talking about. So all of you might have come across this term called hospital-associated infections. We often see that people who go to the hospital for a different condition, maybe a small surgery, if they catch an infection in the hospital, it becomes very difficult for us to treat those infections and for the person to fully recover from such an infection. So in that sense, hospital-associated infections are typically infections that you catch in the hospital. Right? So who's the target group? You see that neonates, which are newborn infants, or patients who are undertaking surgery, who have a very poor immune system, who have an immunocompromised state. These are individuals who are more susceptible to these infections. And once the person gets the infection, the issue is that they end up having to stay in the hospital for a long time. there's a very high amount of mortality and morbidity, which might even sometimes lead to loss of limb or loss of an organ in itself. So all this together actually contributes a lot to the burden, disease burden that countries face. So if you look at these graphs on the right side, what you're essentially seeing is basically the incidence of hospital-associated infections in high-income countries versus low-medium-income countries. So our country would fall under the second category, where you can see that the disease burden of overall hospital-associated infections is about twice that of what we see in high-income countries. And this high incidence of hospital-associated infections in low and medium income countries actually brings down the economy of the country substantially, because there's a lot of cost associated with getting rid of these infections once we catch them. And India used to be famous for what we call as medical tourism. So you used to hear that a complex surgery was performed for an international patient who arrived, and the treatment was given and they went back healthy, right? But now that sector, the medical tourism sector, is itself under threat because of the hospital-associated infections that we have. So a lot of bacteria do cause hospital-associated infections. We are aware of the ESCAPE group of pathogens. ESCAPE is an acronym. If you look at the slide here, ESCAPE is an acronym for all the bacteria which are known to cause hospital-associated infections, including E. coli, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterococcus. Right? So it's an acronym for all the bacteria that are normally causing hospital-associated infections. Now, why am I interested in Klebsiella among all of the other microbes? Because E. coli is well studied, Staphylococcus aureus is quite famous for causing a lot of nasty infections. But the problem with Klebsiella is that it is relatively understudied, meaning there are a handful of groups, about just 5-10 groups in India, and across the world is now picking up pace with Klebsiella pneumonia research, whereas in India, the number of people who are actually studying Klebsiella is relatively less. And why should we study Klebsiella? Because in the hospital setting, it is known to cause infections of the respiratory tract. So typically there's a condition which we call as ventilator-associated pneumonia, where anybody who's put on the ventilator can catch pneumonia, an infection from the ventilator, and then the person goes into pneumonia, cardiac arrest, and death. So in the same context, anything that involves putting a medical device inside you, for example, a urinary catheter or a bloodstream catheter, whatever, if it's a medical device that's going into your body, there's a chance that you might catch an infection from it. Very common infections that we see for Klebsiella are urinary tract infections, where indwelling catheters are put into patients when they come in for surgeries or treatment, routine treatments. In addition to that, we also see that a lot of infections come from surgical sites and wounds. Pus is a very, especially in the context of diabetic foot ulcers, where we see a lot of Klebsiella infections. And the final type of infection that I want to talk about is bloodstream infections. Now, this is very common in neonatal settings where newborns catch this disease typically from the healthcare setting, right? Like from the hospitals or from the caregivers, nurses, anybody could be the source of this infection. Now, these are hospital-acquired infections. Right? So on the right side, you're seeing that it can also cause certain other invasive diseases like gut colonization, diarrhea associated with Kleb, pyelonephritis, which is basically an abscess formation in the kidneys, pyogenic liver abscesses, which is again an abscess in the liver, endocarditis, where it colonizes the walls of the heart and tries to cause an infection there, meningitis, infection of the brain tissue, endophthalmitis. This is again very common, an infection of the eye in Klebsiella, bone infection, which is osteomyelitis, and diabetic foot ulcers. Now, when you look at the type of strains which are actually causing hospital-associated infections versus your invasive infections, there's a very clear difference. So Klebsiella is slowly evolving to be a community-acquired pathogen rather than a hospital-associated pathogen. Earlier, it used to be a hospital-associated pathogen, meaning you don't have a high chance of catching Klebsiella if you don't go to hospitals frequently. But now it's becoming community-acquired. This is because Klebsiella is slowly developing virulence properties, which is enabling it to cause disease even in healthy individuals. Remember when I said that Klebsiella normally causes infections in people who have a suppressed immune system. But the new strains of Klebsiella, which are emerging today, are able to cause infection even in a healthy individual. Now, this kind of infection is actually something that we really need to be worried about. To complicate matters, Klebsiella is also accumulating a lot of antibiotic resistance. So I'm sure you must be aware of drug resistance, wherein you have a lot of abuse of antibiotics in the form of, not following doctor's prescriptions, or sometimes even using antibiotics as growth promoters in the poultry industry. There are a lot of causes for antimicrobial resistance, but what you're seeing here basically is the category of antibiotics with the resistance map, where the percentage of invasive isolates that are resistant in terms of Klebsiella. So we see that across the globe, Klebsiella has a greater than about 60% resistance to all commonly used antibiotics that are currently in use today, right? So around 60% of resistance to aminoglycosides, carbapenems, cephalosporins, piperacillin, tazobactam, even fluoroquinolones. 69% resistance. And you can see India over here somewhere in the middle, right? But if you look at polymyxins, these are basically last resort antibiotics. So if nothing else works, that's when the doctor is going to prescribe polymyxins for you. And even for polymyxins, 15% of the strains are resistant in India, which means essentially for those 15% of the strains, if somebody happens to be infected by those 15% of the strains, there is virtually no treatment. And how is this possible that Klebsiella is actually accumulating all of this drug resistance? It's because of very high levels of horizontal gene transfer. Sure, you must be knowing horizontal gene transfer, conjugation, transduction, and transformation. Through all these mechanisms, Klebsiella has kind of developed a very, very plastic genome with a lot of recombination, integration of drug resistance genes, integration of new virulence genes. And because of this, it's becoming very, very highly difficult to treat, as well as has a very high mortality, which means more than 60 to 70% of people who are infected with the hypervirulent strains end up dying. Now, the requirement for us to manage this difficult situation is to have a method to track the spread of virulence and drug resistance, right? So conventionally, this is done through laboratory tests, which are highly time consuming, as well as, a lot of effort consuming. So the answer to this problem could be what we call as genomic surveillance, which is where we sequence the genome of the microbe to understand about its spread ability, its virulence, its drug resistance, and we also try to even tailor or personalize our treatment method so that the patient recovers quickly, right? So I've told you enough about why Klebsiella is important, because it's gaining antibiotic resistance and it is becoming highly virulent. So the question that we should ask next is, what makes Klebsiella virulent? Why is it becoming more virulent now, right? Remember, I told you there are two types of infections. One is hospital associated and the other one is community acquired. Now, if it's moving from hospital to the community, it means that it's becoming more virulent, right? More ability to cause disease. Now, it turns out Klebsiella has a lot of virulence factors which help it cause the disease, right? So what are these virulence factors? Now, what you're looking at here is classical Klebsiella, which causes infection only in the hospital. So classical Klebsiella has these basic virulence factors, which include the capsule, which is a polysaccharide layer, which protects the bacteria from the host immune system. It also produces siderophores, which are systems that are in place to capture iron from the human body and supply it to the bacteria. It also produces type 3 and type 1 fimbriae and lipopolysaccharides. Lipopolysaccharides offer a certain level of defense from the host immune mechanism, and type 1 and 3 fimbriae are important for attachment to surfaces. Remember when I told you that it's Klebsiella usually attaches to medical devices. Now this attachment is enabled by type 1 and type 3 fimbriae. Capsule protects it from the environment. So with all of this, it becomes very easy for the bacteria to survive in the immunosuppressed host in the hospital, right? But now what has happened basically is Klebsiella has evolved into hyper-virulent Klebsiella, which basically has undergone certain changes. Now, what changes are these? The capsule level that it is producing has increased substantially, meaning there is a much thicker wall for us to break before we can reach the bacteria. If you use antibiotics, the concentration on the outside wall of the antibiotic of the capsule is not going to be the same as on the inside. So antibiotic concentrations will have to be increased. Furthermore, the immune cells, to reach the bacteria, they have to breach the capsule and then only they can reach the bacteria. Even the basic complement system, which protects the bacteria, which kind of kills bacteria once it enters the cell, actually requires access to the cell membrane. Now, if the presence of this thick capsule actually prevents this access and thereby making a very mucoid kind of appearance for the bacteria, that's how you see this. This is a string test that's normally done to check if a bacteria overproduces capsule. So in this case, if a string is greater than 5 millimeters, typically that means that it's overproducing capsule. In addition to that, it has also acquired a couple of more siderophores. So here you see that there are only two, Enterobacter and Yersinia bacter. But here you also see that it has taken up salmochelin from Salmonella and Aerobacter, which is again from E. coli. Now, the presence of these two genes actually confers additional advantage for the bacteria that it can now scavenge iron in a much more efficient way, right? Okay, so given this introduction, I'm going to jump into the work that we do. So this work was started just about a few months back, around 6 to 8 months back, when we collaborated with our sister institute here. We have an All India Institute of Medical Sciences, AIIMS, in Jodhpur. And we collaborate with the microbiology department to offer our services in terms of genomic surveillance. So what we do is we collect strains that are important and relevant, and we sequence the genomes, we analyze, and we try to give them feedback on what can be done to avoid such strains from getting into their hospital as well as into the community. So basically, we look at what are the characteristics of these strains. Typically, when you go to a microbiological laboratory, the only thing that they check for is to try to identify the microbe, what is causing the infection, and to tell the doctor how to treat the infection. People look at antibiotic susceptibility profiles, which will tell them kind of what drugs to use to treat this kind of infection. But we do a little bit more extensive characterization of the strain, where we do a lot of experiments to figure out the basic understanding before we go into genomics. So basically in our lab, we try to look at antibiotic susceptibility of the bacteria. So this is done in collaboration with AIIMS. So they use the Vitek2 device, which is an automated device which can actually identify the bacteria and can also tell us what antibiotics it is susceptible to, what is the concentration of antibiotics to use for treatment. So this is on the medical side, and they use this device routinely to test for antibiotic susceptibility. On our side, we try to test for siderophores, the ones that I just mentioned. What kind of siderophores does the bacteria produce? Is it mucoviscous, meaning does it produce a lot of capsule? We also do an assay to test for how much capsule the bacteria produces. And we also perform what is called as a serum resistance test, which is a proxy to test for the capsule as well as the lipopolysaccharide production of this strain. So all this together will tell us if a strain is virulent or is it hypervirulent, right? And once we have this information, then we go about doing our genomics. So I'll just walk you through our findings from the last few months. So this is a typical Vitek report. This is how it looks like. So I'm just showing you data for about 6 strains that are highly interesting and would be kind of interesting to follow. So the U strains are typically isolated from urine, meaning they cause urinary infections. And the P strains are isolated from pus, typically any abscess or any fluid, any wound, for example. And the BC strains are isolated from blood cultures, right? So these are bloodstream infections. So if you look at it, as you would expect, there are some strains that are highly sensitive to antibiotics, like for example, U233 happens to be sensitive to most of first-line antibiotics. You don't even have to worry about second line, so the testing is not done. But if you look at U23 on the other hand, which is a urinary isolate, it's extremely resistant to almost all antibiotics except colistin and is intermediate for tigecycline. So it's resistant to gentamicin, ciprofloxacin, cotrimoxazole, everything, you name it, it's resistant. All carbapenems, even meropenem and ertapenem, right? So if you look at it, most of the bugs are actually resistant to most antibiotics. We do have a couple of strains which are sensitive. And it was by choice that we picked up these strains for sequencing, because we wanted to kind of see what kind of virulence factors are present in the drug resistant versus the drug sensitive strains as well. Now, moving on, we also test for production of siderophores. So in all our assays, we use appropriate controls where we pick up standard ATCC cultures, which will serve as control. So here 13883, 1706, 2146, and 14330 are all standard cultures. So you can see that they produce low to moderate amount of siderophores, but we also pick up some strains which actually produce very high amounts of siderophores, like R65 and R30. Some strains are in the intermediate, like the BC strains, which are BC 135, 716, and 387. So this kind of tells us that a bacteria produces siderophores or not. To a semi-quantitative extent, we're also able to see that some bacteria actually overproduce siderophores as compared to the other ones. So the next study that we do is we look at the capsule production ability. So if you recall, I mentioned that, let me go back to that slide. So I was talking about the string test here, which will tell us if a bacteria is mucoviscoid or not, if it produces a lot of capsule or not. Now, the threshold for this test is actually 5 millimeters. But in reality, we have a couple of isolates which are producing strings that are around 7 centimeters long, right? So 5 millimeters is the cutoff, but we're looking at strings of about 7 centimeters long. So we are seeing a very high level of mucoviscosity, at least in one of the strains that we have actually sequenced. There are a few more which are in the pipeline. So P34 is one such strain which produces a 7 centimeter long string test. So this is a very easy test to do. You just grow the bacteria on the medium, you touch it with a toothpick or a wooden applicator, and when you extend it, a string forms, a complete string forms. And if it is longer than 5 millimeters, you can classify it as a string test positive bug. So this has to be actually cross-verified by another assay, which we did, which is called as a sedimentation assay, where we just centrifuge the culture at low speed, and then we look at the supernatant and the pellet. We try to see the difference in optical density. That will tell us whether a bacteria is capable of pelleting down during low speed centrifugation. So the understanding is that if a bacteria has a very thick capsule, it doesn't really pellet down that efficiently. So P34 turns out also has a very high level of mucoviscosity, while all the other strains that we tested were about 0.3 to 0.4 was the maximum. P34 had a mucoviscosity of about 0.8. We also went ahead and did a capsule polysaccharide assay for all the strains that we are currently exploring. So here, basically, we do what is called as a uronic acid assay, where we extract the capsule or polysaccharide of different bacterial strains, and we assay it using standard methodology to assess how much uronic acid is present in the capsule. So it's kind of an indirect measure of how much capsule a bacteria produces. Now, surprisingly, if you look at the previous slide, P34 clearly stands out as a very high mucoviscous strain, but on the capsule side, it's actually much lower than the other strains that we have tested. So P34 does not appear to be producing a very high amount of capsule. And I'll walk you through it when we look at the genome as to why that might be actually happening. So this report actually is where we do a human serum susceptibility test to check for its ability to tolerate human serum. So basically, human serum contains active compounds like the complement system, which will form a pore-forming complex on the surface of the membrane of the bacteria and degrade the bacterial cell membrane, right? So if you expose bacteria to serum, you expect the bacteria to die, right? But unfortunately, what we see is most of the strains that we tested, if you look at BC387, 135, 716, all of these are resistant. So on the left, you have plus serum, on the right, you have minus serum. So in both, the counts are almost identical, which means the presence of serum has no effect whatsoever on these bacteria. On the other hand, if you look at the reference cultures that we used, which are ATCC cultures, you see a substantial amount of killing through serum effect, right? So when you add serum, the number of bacteria decreases as the dilution increases. Here it doesn't, it's somewhere intermediate. So basically you would classify this as intermediate because there's some killing, but it's not completely eliminated. This P34, the one that I was talking about in my earlier slide, is completely sensitive, meaning all bacteria are completely killed by the presence of human serum, which is kind of controversial because if you have mucoviscosity, bacteria is expected to be resistant to serum. But our strain is actually sensitive to serum, which is kind of interesting. And that's why we're doing genomics with P34 at this point of time. We also found several strains that are highly resistant to serum, like for example, U233, which is actually an antibiotic sensitive strain. So if you think about it from the bacterial context, it kind of makes sense. If you have a very thick capsule, which helps you resist serum, it does not matter if you have very high concentrations of antibiotics. So you don't need genes that are required to detoxify the antibiotics, right? So in that sense, it kind of makes sense. So U233 and U23 are quite resistant to human serum as well. So let's move on. I just walked you through some of the basic experiments that we do so that you get an idea of what we actually do. So with this kind of basic strain characterization, we then move on to the genome sequencing of the bacteria. So I want to introduce you to this term called genomic surveillance. Although most of you would have heard about it, given the current situation that a lot of genomic surveillance is being done for COVID-related situations, right? So basically, genomic surveillance is nothing but sequencing of the genome to assess if an infectious agent is present. If it is present, how is it evolving? Where is it spreading? And how is it changing as it moves from one person to one person? For example, this is patient 0. Now, let's say this person passes on the infection to about 5 people, and this person passes on the infection to this. So when the transmission happens from patient 0 to these 5, what was the genetic structure? And then when the next shift happens from this person to the next person, what changes have happened in the genome? And so on and so forth. So as the transmission happens, the gene sequence changes because the bacteria also kind of tries to adapt. Now, this is very common in the Klebsiella setting. We have seen, we've heard from our clinician collaborators that even when one person is being treated with antibiotics, we see the evolution of resistance to antibiotics during treatment. For example, the bug might have tested sensitive for a particular antibiotic. The doctor would prescribe that antibiotic, but during consumption of that antibiotic, the bacteria becomes resistant to that particular antibiotic. So this is pretty common, and that has to happen through genetic changes. And we are interested in identifying what are the genetic changes which are actually causing resistance to certain antibiotics. Now, this is in the context of the COVID situation. So let's not read too much into it. So in the global setting, a lot of work has been done for Klebsiella, where people are looking at a lot of genomic epidemiology where different clonal groups have been characterized in terms of the presence of different virulence genes, absence of virulence genes, as well as the presence of drug resistance and determinants as well. But in the Indian context, we don't know much because there's very poor availability of data as well as analysis ability of data. So right now, what we're doing in my lab is we're looking at around 1,000 Klebsiella genomes in the context of drug resistance, as well as in the context of virulence factors, to see what kind of strains are here, what kind of drug resistance profiles do they have, what kind of virulence factors do they carry, right? So in this context, I would like to introduce Oxford nanopore sequencing. So this is a very fun kind of experiment that we do very frequently in the lab. Basically, the nanopore sequencing technology works based on a nanopore, which is basically a protein pore, which is kind of fixed on a membrane, right? So there's liquid on both sides and the current is applied. So you have DNA on one side, and as the DNA moves through the pore, there's a helicase sitting on top, which kind of unwinds the DNA. So one strand passes through the nanopore. And what the technology behind this is that they have developed a strategy to kind of measure the current within the nanopore chamber as the DNA passes through it. So when there is no DNA, they would develop what is called as a baseline signal. And as the DNA enters the nanopore, this baseline signal gets changed, right? So, and it has a very unique signature for every base that is passing through the nanopore. So as the DNA passes, they're able to decode the sequence of the DNA based on the change in the current, the signal that's actually generated from the nanopore. Now, the biggest advantage of this method is that it's a long read sequencing technology, meaning it can generate no extremely long number of reads because there's no synthesis step involved. And it's also very economical in terms of time for library preparation. So the workflow is as follows. We collect the strains from AIMS Jodhpur. We characterize them first, and then we isolate DNA. We prepare library, then we sequence on the nanopore. I'm sorry, I forgot to introduce. So this is how the nanopore sequencer looks like. It looks like a slightly big pen drive. Costs about 1.5 lakh rupees to set up the sequencer. I would highly recommend it if you wish to train your students on sequencing and analysis of sequencing data. It's a really fascinating equipment to have if you guys want to explore sequencing a little bit. So once the sequencing is done, we would base call the data, do quality control, perform a long read assembly, polish the assembly, do a contiguity check, and then do annotation. So annotation is the process wherein so basically assembly is where the different fragments of DNA that you see here, it's not a continuous stretch of DNA, it's a fragmented DNA. So these fragmented DNA are put together in the form of an assembly, and then we annotate it. We try to say, okay, this section of the gene corresponds, or this gene corresponds to this function, right? So once annotation is done, we then do a lot of comparative genomics where we try to align the genome with a lot of other genomes that are already available in the database. We do plasmid analysis, what kind of plasmids are present, what kind of drug resistance genes are there, what virulence factor genes are there. And for all of this, we have developed kind of pipelines, and we also use pipelines that are already available in the public domain. So moving on, I'll give you a snapshot of how the genomics of the same strains that we looked at in the characterization looks like. So P34, the strain with the long string, right? So this was isolated from an adult, from a fluid. It had one contact, meaning there's no plasmids, right? There was no plasmids in this strain. It's important to note here that usually hypermucoviscoid strains, or strains which have these long strings, are typically, they have a virulence plasmid. Now, what is unique about this strain is it does not have any plasmid, and yet it is hypermucoviscoid. So we're currently looking at what are the genetic mechanisms that is enabling the bacteria to be hypermucoviscoid without the presence of a plasmid, right? So here you have the number of genes listed, number of protein coding genes, number of RNA coding genes. And this is how it looks like. The overall picture looks something like this. So you would see that the urine isolates, again, if you recall, U23 was the culture which was highly drug resistant. And as expected, there are 9 plasmid sequences detected here. And U233 is also a partially drug resistant strain, but there's no plasmids. So P34 and U233 do not have any plasmids, and the other strains all carry plasmids. I'm emphasizing on plasmids because plasmids are one of the largest vehicles of horizontal gene transfer. Let me just pause here for a minute to ask the organizers, it's already lunchtime, do you want me to continue or should I stop now?
Thank you so much, sir. Participants, do you have any questions?
Ms. Uma, I'm not yet done. I'm just asking, do you want me to stop here or do you want me to continue?
Shankar, you can continue.
Okay. I was just wondering because it's already lunchtime. Okay, so I'll be quick. I'll just try to finish it very soon. So, so basically, we have the genome sequence of about 50 in our lab, and I'm just showing you 6 for representational purposes. So we selected some strains from the public database for analysis. So we took strains from all across India, with about half of the data, about 400 strains we have taken from, most of it came from Bombay, rest of it came from the rest of the country. So what we did basically was, the first step that we did was to categorize our strains based on what we call as multi-locus sequence typing. So basically we look at different highly conserved loci on the genome of each of these strains, and we try to place them phylogenetically. We try to put them on a phylogenetic tree, if you will. So basically U23, the strain which carried a lot of plasmids and was very highly drug resistant, all of the types, all of the strain, right? We were not able to place them into a known sequence type. So this means that it is possibly a new lineage of strains which is causing infections in Western India at this point of time, especially in the hospital setting. So all the strains that we have tested are either single or double locus variants when compared to known strains. And we are currently looking at what kind of genomic rearrangements have happened in these strains, so that we'll be able to better understand how the strain is evolving at this point in time. Now in India, if you remember, I told you we analyzed about 400 publicly available sequences. If you look at the sequence types, most of it is from ST14, some of it from ST147, and most of it from ST231. Right? So these are the three major. So these are the three major sequence types that are commonly found or known to be present in India. Right? But the ones that we actually identified are in fact close to ST16. So it's an unknown sequence type, but these isolates are actually close to ST16 and ST11, which are global high-risk clones, meaning these have been declared as clones of very high risk in the global setting. Just to illustrate my point, we did construct a phylogenetic tree. So it appears that U233 happens to be on a completely separate node without similarity with any other strain. And the closest similarity that we could find was to ST320, which was again completely different. In a similar manner, if you look at P34, it also forms an independent branch, meaning that it's not really close to any of the known strains. But U23 and R62 and a few other strains that we discussed about are pretty much close to ST16 or ST15 in this case. Right? So these are again globally high-risk clones. In terms of drug resistance, we found an enormous number of drug resistance genes, both on the chromosome as well as on the plasmids. Especially if you look at U23, it has a high number of plasmids. BC strains, the 135, 387, and 716 also do contain a lot of things, but you can actually see a very nice pattern here. You see that the beta-lactamase SHV is almost always present chromosomally. And we also notice that the other beta-lactamases are coming from virulence plasmids or drug resistance plasmids, such as the TEM or the CTX or NDM. NDM, I'm sure you guys must have heard of, because this is one of the very famous New Delhi metallo-beta-lactamases that are isolated from Indian strains. Now, one more interesting observation that we made was for U23, we also detected this BLA-CTX M15 on the chromosome. Now, BLA-CTX M15 on the chromosome is very rare. This is because typically beta-lactamases tend to be on plasmids. Now, what has happened here in this particular strain is that from the plasmid, it also has a copy of beta-lactamase CTX M15 on the plasmid. So it appears to us that a jump has happened from the plasmid to the chromosome, meaning that now this BLA-CTX-M can be vertically transmitted from U23 to other strains, which is a serious public health concern in India. We also looked at what kind of virulence factors are present in Klebsiella. If you remember in these strains that we have, that I'm showing you right now. So if you recall, I told you that to attach to medical devices and to attach to tissue, it requires a lot of adhesins, which are called as type 1 and type 3 fimbria. So if you look at type 1 fimbria, we located almost all the genes in all the strains, except in FIM-A, which basically is absent in the urinary isolate. So basically, we're looking at what kind of virulence determinants are present in which samples. So for example, are the virulence factors different for causing a urinary tract infection and for a bloodstream infection? That's what we're interested in asking. In a similar manner, we also looked at the siderophore production. So again, we were surprised to see that none of the strains had the ability to synthesize aerobactin or salmocylin for that matter, but they both had the ability to take up aerobactin and salmocylin from the environment, meaning these are examples of xenosiderophores, meaning a bacteria does not produce its own siderophore, but it can actually capture siderophore from other bacteria and utilize it efficiently. And just to make a point here that siderophores are much more common in hypervirulent Klebsiella. So the presence of aerobactin is actually dominant in hypervirulent Klebsiella. So this is actually a graph where you see that this is, classical Klebsiella, where you see there's not much of siderophores. Here you see that there's actually a lot of siderophore presence, right? Amount of siderophore that's being produced. We also noticed a pattern in terms of which isolation source has what kind of siderophore. So for example, we noticed that in the case of blood isolates, we saw that the bacterial production was predominant, whereas in the other sources, it was actually not there. And we also noticed one more very interesting aspect, that there was a pyochilin transporter or a receptor. So pyochilin is basically a siderophore which is actually produced by Pseudomonas, not by Klebsiella. So Klebsiella has somehow evolved the mechanism to capture the siderophore of Pseudomonas and use it, right? So that's a very interesting finding that we have made. In terms of capsule, we did notice that there were a lot of genes, all the genes required for capsule synthesis was present. Unfortunately, although we had one strain which was actually hypermucoviscous, which means overproduction of capsule, none of the strains actually had the genes that are required for overproduction of capsule. Usually in hypervirulent strains, RMPA is a gene which is expected to be there if a strain is hypervirulent. But in P34, we do not see this. So what we are proposing right now, we have a model that P34 is basically a new capsular type, which has a novel mechanism for hypervirulence, as opposed to other strains that are already known. fact, we've identified 8 new genes in capsule in P34 capsule region, which we are characterizing genetically to understand what kind of hypervigilance or hypermucoviscosity is. Interestingly, we also noticed that on the genome, find a genomic island, which is also happens to be in the capsular locus of P34. Now, secretion systems are extremely important in Klebsiella because these are important for the bacteria to compete with each other. For example, Klebsiella is a commensal that lives in human gut. So in the human gut also, it kind of competes with other microbes. So for that purpose, it's required for Klebsiella to be able to inject toxins into other bacteria. And type 6 secretion systems do exactly that. So it's basically a spike-based injection protein, injection complex is made. So interestingly, in almost all the strains that we have tested, we found that most of the strains carried genes that are required for construction of this type 6 secretion system, which means that if you have Klebsiella in the gut, it's going to actually influence the gut microbial community. So to wrap up, I would just like to summarize quickly. So we've identified a lot of unknown sequence types of Klebsiella that were circulating in the hospital environment. We have also concluded that 3 of these strains that we have sequenced are actually just a single outbreak strain. So let me just go back and show you here. So if you look at these 3 cultures, they cluster together, right? BC387, BC135, and BC716. Turns out they are all the same strain, but it has actually caused 3 separate infections in the neonatal ICU at AIIMS Jodhpur. So we are currently working with AIIMS to see how the strain has originated, and we're looking at the patient histories to understand what kind of antibiotics were given to these patients and how, if they recovered or not. So overall, we would like to conclude that genomic surveillance using nanopore would be able to serve as a gold standard for molecular epidemiology. We would be also possibly able to develop more specialized and personalized treatment plans for highly drug resistance genes. And it's also easy for us to track the spread of virulence factors and antibiotic resistance mechanisms in Klebsiella by nanopore sequencing. In the long run, we hope to be able to develop vaccines against these high-risk clones so that people who go to the hospital can take these vaccines and prevent infection during the hospital stay. As I reach the end of my talk, I would like to thank the organizers of this conference for inviting me to be a part of it. Special thanks to Dr. Ilakyam for allowing me to be a guest of this conference. Also thanks to the Vice Chancellor of the university, as well as the Registrar and the Head of the department for allowing me to be a part of this conference. I would like to acknowledge the TPT Wellcome Trust India Alliance for funding, IIT Jodhpur for infrastructure, and my collaborators in AIIMS Jodhpur in the Department of Microbiology. So all the work that I have presented today is the work of my students, Ardendu, Asta, Lavanya, Tamal, Sharvika, and Anuja, who do all the hard work. I just try to mentor them and guide them. So with that, I would like to thank you for your attention. And if there are any questions, I'll be happy to answer. Before I close, I just want to inform that my lab also works on the human gut microbiota, where we study the effect of human gut, different drugs on the human gut microbiota and how it actually influences health and disease. Actually, there was a question in one of the earlier sessions where there was a question about how the gut microbiota can be used for forensic analysis. So I would like to just address that question. So basically, the normal gut microbiota that lives in our intestines will change every day after the patient has died. So from the gut microbiota, it's possible to determine the time and date of death depending on the community structure. So that's how it's actually influenced. You can use it in forensics in terms of calculation of the death time of an individual. Also, the microbiota on your fingertips can actually be used easily to track if it is actually you who was present in a crime scene. So microbiome can also be used in forensics. With that, I'd like to thank all of you again for your attention. And if there are questions, I'll be happy to answer. Thank you.
Good afternoon, sir. I would like to ask one question, sir.
Thank you so much, sir. Participants, any queries?
Yeah, please go ahead. Did anybody have a question?
Yes, sir. I have one question, sir.
Yeah, please go ahead.
Sir, basically you have talked about Klebsiella pneumoniae. So I want to know that how sir, basically you have talked about the Klebsiella pneumoniae. So I want to know whether the symptoms and window period of this pneumonia differ from other pneumonia caused by Streptococcus species or some other virus like SARS, or it is same as that.
Klebsiella pneumonia is a bacterial infection, so it's completely different as you would expect from other viruses or other bacteria for that matter. So it's definitely different. Yes, the window period
sir,
also.
window period would, okay, sir. Okay, thank you so much, sir.
Yeah.
Thank you, Pranjal. Sir, thank you so much for such a nice presentation, sir. And in the present scenario where multi-drug resistant bacteria are a major hindrance in the pandemic around, your talk was very informative for us, and you told us about the Oxford nanopore sequencing technology, and it was very new for us. You told it was fun to work with that. So we hope one or the other day we'll be there to work with that, sir. Thank you so much, sir. Joining with us in spite of your busy schedule, sir. We have the organizer to thank you also, sir.
Thank you so much, Shankar.
Thank you. Thank you so much, ma'am, for your invitation.
Thank you so much for sharing your research work with us. Thank you so much, Shankar.
Thank you. Thank you, ma'am. Thank you. Thank you all.
Thank you once again, sir. Dear participants, we are moving for lunch now. Kindly join us again at 2 o'clock for the next session. Please do remember the time is 2 o'clock. Please join us for the next session at 2 o'clock. Thank you.
Srini.
Yes, sir.
Good afternoon, Srini. How are you, Srini?
Yeah, good afternoon. How are you, sir?
Good. So morning I was attended our Dr. Professor M.S. Reddy sir lecture also. Inauguration as well as. It was excellent lecture. Morning he mentioned about you.
Okay.
And one of my best entomologists ever come across in the world. So he's going to give this lecture for vegetables like that. In these topics, some vegetables under the okay.
Okay.
Situation
it's all going fine, sir. Now the situation is under control.
Okay.
So far it's
Philippines was the neighboring country, Taiwan.
ஆமா பக்கம்
தான் அங்க வந்து ரொம்ப கொஞ்சம் சீரியஸா இருக்கு per day வந்து 10,000 report ஆகுது இருக்கு.
ஆமா
Just now I spoke to Dr. Jawahar Ali. He's in, you know him?
I heard about him.
He's a hybrid rice breeder based at Philippines, Hiri.
Okay.
அவர் கொஞ்சம் விஷயமா பேசிட்டு இருக்கும்போது சொன்னாரு.
Okay.
ஒன்ஸ் வந்து இங்க ஏதாவது ஒரு விஷயத்துக்கு வந்தாச்சுன்னா that visa will be invalid. திருப்பி போக முடியாதாமே?
Yeah, that's the situation now in many countries.
Yes. நம்ம டாக்டர் செல்வாவோட அக்கா பையன் செக்கோஸ்லோவேக்கியா இருந்து வந்து he lost his job. He could not able to go back.
Oh my god.
ஒரு 7-8 months பார்த்தாங்க. After that they canceled his visa.
Okay.
Now he is suffering. He is looking for some job in India, Tamil Nadu. அப்புறம் ஃபேமிலி எல்லாம் இருக்காங்கல்ல இன்ஜெக்ஷன் எல்லாம் போட்டீங்களா வேக்சின்?
நான் போட்டுட்டேன் சார். இப்போ ஒரு 2 weeks ஆகுது.
என்ன ஃபைசர் போட்டீங்களா?
மாடர்னா போட்டேன்.
மாடர்னா ஓகே வெரி குட்.
வைஃப் வெயிட் பண்றாங்க. ஏன்னா 2 பேரும் ஒரே டயத்துல போட்டு ஏதாவது எஃபெக்ட் இருந்தா என்ன பண்றதுன்ட்டு.
சரி. இல்ல ஒண்ணு இல்ல இப்ப சீனிவாஸ் போடலாம், ஒண்ணும் ப்ராப்ளம் இல்ல.
ஓகே.
இந்த ஒரு 2 டேப்லெட் போட்டோம்னா ஜஸ்ட் ஒரு 1 day 1 or 2 days ஜஸ்ட் ஒரு ஃபீவர் லைட்டர். அப்புறம் நம்மளோட இந்தியன் ஆபரேஷன் எப்படி போயிட்டு இருக்கு?
It's going fine, sir. Now I don't focus much on the South Asia region because it is a separate regional director.
Based at who is that Nair?
Dr. Nair, Ram Nair.
Okay. அதனால அதுக்கு அதிகமா கான்சென்ட்ரேட் பண்றது இல்லையா?
ஆமா சார்.
நம்ம ரவிசங்கர் அங்க இருக்காரு.
ரவிசங்கர் is there. ஆமா.
இல்ல நம்ம லாஸ்ட் டைம் நம்ம டிஸ்கஸ் பண்ணது மாதிரி கொஞ்சம் அவங்க சொல்லுங்க, எனக்கு ஒரு வாய்ப்பு கொடுங்க. அந்த
ஆமா நான் சொல்லிருக்கேன். நீங்க you talk to him. You talk to Ravi.
ரவியோட நம்பரை கொஞ்சம் ஷேர் பண்ணுங்க, எனக்கு இருக்கான்னு தெரியல, செக் பண்ணனும்.
Okay. I will send it to you. அவங்களும் எல்லாமே இருந்துட்டு இப்பதான் ஒரு 2 weeks தான் கேம்பஸ்க்குள்ள போயிருக்காங்க because they allowed like 30% staff, 60% staff.
Okay.
Yeah.
Okay. I think cost has come.
இது இவங்க என்ன ப்ரோக்ராம் they stick to the schedule சார்?
ஆமா இப்போ வந்துருவாங்க. கால் பண்ணிட்டுமா? just a minute. அந்த வந்துட்டாங்க சிஎஸ்காம்.
Okay.
Good afternoon all. Hope you had a good lunch and welcome back once again to the ICABB-2021. So we've been having wonderful sessions today and now it's afternoon and we have a special guest with us as a resource person for this session. Along with us, we have Dr. Srinivasan Ramasamy sir. Sir has received a PhD in agricultural entomology from Tamil Nadu Agricultural University and Ancient Vegetable Research and Development Center, Taiwan in 2003. And from 2005 onwards, sir has served as the head of entomology group and currently leading the flagship program for safe and sustainable value chains. He serves as a chair of the World Veg Research Management Team. Sir has obtained more than 28 million USD for research and development projects as project manager of the flagship program, and Sir has coordinated research and linkages in more than 20 countries in 6 continents. Partnerships include universities, private sectors, pharma groups, NGOs, governmental and international organizations, and Sir has supervised more than 75 students, including postdoctoral fellows. Sir has also offered several training programs for farmers and other groups. And Sir is an active member of many professional societies, and including Asian PRP Society of Sustainable Agriculture. Sir has published more than 170 papers, and one of the notable publications is a review article on Maruca vitrata published in 2021. And to add to his crown, Sir has also received several awards, including the International Plant Protection Award of Distinction, and ISHS Medal, and Veginet Sewage 2019 Award. Sir, we are absolutely delighted to have you with us, sir. Over to you for the session, sir.
Okay, thank you very much. First of all, I would like to thank the organizers of this international conference for inviting me to deliver this talk. Let me share my screen. Okay, is it visible?
Yes, sir, visible, sir.
Okay, thank you.
Go for
okay,
slide mode. Srini, please go for slide mode.
Sorry?
Go for slide mode.
Yeah, I have gone to the slide mode.
Fine, okay, fine.
Okay. So I have the pleasure of giving this talk on the transformation of food systems for food safety as well as nutritional security, with a special emphasis on vegetables. Probably some of you might be aware of this gentleman whose name is Paul Hermann Müller, who is the developer of the very popular pesticide dichlorodiphenyl trichloroethane. In short, it's called DDT. Even for this discovery, this guy was awarded the Nobel Prize in Physiology or Medicine in the year 1948. You know, just during the World War, many of the soldiers were affected by the malaria, which was transmitted by mosquito. So the insecticide property of this particular compound was identified by Paul Müller. So it was used in the control of mosquitoes as a major drive to prevent the malarial disease. So after the World Wars, there was a huge amount of DDT, and the scientific world was turning its attention towards agriculture. So they rediverted the DDT for the management of agricultural pests. So this is one of the notable discoveries in the history of agriculture. And then almost after 22 years, another gentleman named Norman E. Borlaug was awarded the Nobel Peace Prize in the year 1970. And there is no Nobel Prize for agriculture, so he was awarded in the category of a Nobel Peace Prize. You may be wondering why he was awarded the Nobel Peace Prize. He started working in Mexico in 1940s and 1950s. And through his work, he developed a very robust variety of wheat. So in 1956, this new variety was able to provide a very high yield, so that it makes Mexico very self-sufficient in terms of food production. So it was exactly the same time in many of the developing countries in Asia and Africa were suffering from the food insufficiencies. So these wheat varieties were introduced into the countries in Asia, especially in South Asia, including India and Pakistan in mid-60s, so that the productivity of wheat was significantly increased. So the food grain production was significantly increased in those days in this part of the world. So because of that reason, he is considered as the father of green revolution. And because of increased food production, which can alleviate the hunger for majority of the population in the world, which could ultimately lead to peace and prosperity, this gentleman was awarded the Nobel Peace Prize. So this is a brief history in the past. So now coming back to the story of DDT, people were only seeing at the positive side of the DDT, its role in agricultural pest management for another 40 years. So the alarm bell was rang by Rachel Carson, who you see in the screen, who wrote a book called Silent Spring, which depicted the adverse impact of DDT in the environment in terms of its bioaccumulation, its long-term persistence in various elements in the environment, including the soil, water, and also including the food chain. So the reduction in biodiversity, the adverse impact or the pollution being caused by this particular compound, they were all brought to the attention of the mainstream population. So eventually the world was looking for what is the alternative for these broad spectrum chemicals. Okay, so this was a critical challenge at that point of time in agriculture. Is it the only problem challenging agriculture? If you look very critically, there are also a few key challenges for food security. It's not only in 70s or 80s, which is also trailing until today. One of the most significant challenge is the growth, significant growth in the population in the globe. Of course, India is having a very acute problem of the population growth. Then declining cultivated lands, declining agricultural populations, and climate change. Let me briefly walk through all these key challenges. So as I indicated, the world population has grown enormously. So if you look at the next 20, 30 years, almost in around 2050, the population is going to hit 10 billion, the global population. And almost 20% of the population is going to be in one country, that's India. The other problem I mentioned is the declining agricultural land. What we had 50 years ago, proportionately for the agriculture and the human habitat or the industrial purposes, is getting narrower and narrower. So no matter where we are living, be it a developed nation or be it a developing nation, the proportion of land available for agriculture is getting less and less. So just you try to correlate the previous slide. On one hand, we have a significantly increasing population trend. On the other hand, we have a declining area for agriculture. So how are we going to feed the increasing number of mouths in the next 30 years? This is a key question. And another critical problem, I'm not sure how many of you are aware or how many of you seriously consider this fact. If you look at the average age of the active farmer in India, it is 50.1 years, meaning the active farmer in India is at least 50 years or over, meaning our fathers and grandfathers are the active generation engaged in agriculture. So no more the current two generations are actively pursuing agriculture as a profession. Is it a problem only in India? No. You look at the two figures I show here. The one on the left-hand side on the top, which is the statistics from United States of America, where you can see a majority of the farming community falls in the category of 45 to 64 years. So they are also similar to India. And you look at the right-hand bottom picture, which is the statistics from Australia. And again, you can see who is actively farming. It's between 35 to 60 years. So in most countries, no matter whether it's a developed nation or a developing nation, the active farmers are around 40 to 50 years. So if we have a declining youth population who is moving away from agriculture, how are we going to feed the world? This is the biggest challenge in front of us. And comes the climate vagaries. Of course, the scientists are developing high yielding, best performing varieties. But the key question is, are we able to fully realize the genetic potential of these improved varieties? For example, an improved tomato variety may be able to produce at least 70 to 80 tons per hectare. It's the full potential of the crop. But if you take a farmer, say in Salem or Namakkal or Dharmapuri, who is cultivating this particular variety, is able to harvest 20 to 25 tons. So it means, where does the gap of 55 to 60 tons go? It is influenced by multiple factors, including the soil, but the weather plays a critical role. So just you look at this figure. So the yield of the particular corn crop, it is lying somewhere between 60 to 150 bushels per acre. And every time you get a bad weather, be it a blight, be it a drought, or a flood, every time it is pulling down the yield, So now we are living in the era of climate change. So with an unexpected drought, heat, flood, whatever you name it, any sort of acute change in your weather can pull down the potential yield of any improved varieties. Okay, so I was talking about the challenges lying on the production side or supply side of the food chain. Now you look at the demand side or the consumption side of the food crops. There are two challenges we are facing now. One problem is malnutrition. The other problem is overnutrition or obesity. You may be surprised if you see the number of people who are overnutritious than the malnourished population. Just you see this particular page here. On the top, I show the number of people who are underweight, meaning they don't get sufficient calories of food. It's only about 821 million. But you look at the bottom row of this picture, people who are overweight, meaning overconsumption, is almost 2.8 billion. So meaning we are not running short of food grains, but we are running short of the distribution of the food grains in an equitable manner. So the challenge is not the availability of the food. The key factor is the accessibility and affordability to the food. So how are we going to address this challenge? And another critical fact you should understand is the hidden hunger or the micronutrient deficiencies. Sometimes people may be highly ignorant of how much important a particular micronutrient is. If you take half of the women population in India or even in Tamil Nadu, probably half of the women population might be anemic. They might have low iron content in the blood. We may not know the implications of a particular deficiency. I just want to pull out a few numbers for your attention here. You see, each day, 400 mothers die during their childbirth due to iron deficiency. At least 1,400 children go blind because of the vitamin A deficiency. So this is the critical need the society is facing as a major challenge as of today. So how we are going to address these critical issues? So I have highlighted few of the tips of icebergs existing, which are challenging the development of the human capital at this moment. So in order to overcome some of these challenges, there was a global partnership or initiative, which is called Consultative Group for International Agricultural Research, or in short CGIAR, which has come into existence 50 years ago. So across the globe, there are several institutions focusing on the development of different food crops, systems, or the policy side of food production and consumption. So these institutions are located in different countries around the globe under the umbrella of a CGIAR. Now they are making a transformation to an umbrella organization called One CGIAR. So these institutions are trying to address some of the critical challenges I mentioned so far. But these institutions are not holistic enough to address all the challenges. For example, you take the vegetable crops, one of the most important elements in the diets of human population. None of these organizations are working on the improvement of vegetable crops. So because of these sort of gaps, the world was also looking at the possibility of alternatives. So this is where my organization, which is called World Vegetable Center today, has come into existence in the year 1971, exactly 50 years ago, as Asian Vegetable Research and Development Center, in short AVRDC, with a major focus on Asian region. It's a not-for-profit organization, meaning whatever we develop, we don't get the patents, we just share it freely with any sort of stakeholders. So our mandate has been expanded 20 years ago as a global mandate. So we renamed as a World Vegetable Center. So we are working on a mission of doing research and development to realize the potential of vegetables for improving the health and also for improving the livelihood of the population. So we focus on a very broad range of vegetable crops, starting from very popular vegetables like tomatoes, peppers, or beans, onions, cucumbers. But we also focus on the traditional vegetables, which are hundreds of different species, which are most important in the diets or the culinary culture of a particular region or country. So they are called traditional vegetables. Okay, I talk about food systems. You may be wondering, what is a food system? So it's not a very complex concept. It's a very simple concept where everything starting from the food production to the food consumption. So it starts with agriculture in the field, then harvest, processing, and transportation, storage, value addition, and then distribution to the wholesale and the retail chains, and ultimately reaching the consumers for consumption. So all the process along the food value chain is considered the food system. But it's not complete unless we look at the socioeconomic drivers as well as the environmental drivers. Why we need to pay attention to these two drivers? Because the socioeconomic drivers such as policies, the market, they all influence. The best example is, I spoke about the overweight versus malnourished. The food is being produced in sufficient quantities, but the market has not distributed it equally. So the population in one part of the world is having more access to the food, whereas the population in the other part of the world is having less access to the food. So this is where the socioeconomic drivers can play a significant role in terms of making an equitable distribution and making available the food grains equally accessible to all. And environmental drivers. So I talk about climate change. And if you look at the greenhouse gas emissions or ammonia emissions, agriculture, today's agriculture practice has a significant contribution for all these greenhouse gas emissions or ammonia emissions. So unless we change the practice, unless we reduce these greenhouse gas emissions, we are going to exacerbate the problem of climate change more. So we need to make the food production, agriculture, very, very eco-friendly so that it is also compatible with the environmental health. So this is where the environmental drivers plays an important role. So food systems is a combination of food production to distribution combined with its socioeconomic drivers as well as the environmental drivers. So how we can transform the food systems in a way it could be compatible with the environment and also with the society. So here comes a concept, 5 levels of food system change. If you look at the level 1, which is the practice we initiated 40-50 years ago, increasing efficiency of the system. For example, you have a very traditional variety cultivated by the farmer. So the breeders develop a new variety and they try to replace an existing low yielding, disease prone variety. So here we are trying to increase the efficiency of the crop production system. Or I introduce a new pesticide. I mentioned about DDT. So we introduced DDT or similar pesticide for managing the pest. So we are again trying to increase the efficiency. So they are all coming under the level 1, which was the practice in the last century. But as I indicated, once we realize some of the harmful effects of some of these technologies which we introduce, we try to develop alternatives as the substitution. So it has become level 2. For example, we try to develop the biological pesticides or a biocontrol approach that can replace the harmful chemical pesticides. So the biopesticides or biological control becomes a substitution for the harmful chemical pesticides. So in the last 30 years, there was a lot of focus on developing these substitutions or alternatives. So we were also contributing for level 2 in the past. But as of now, the world is also trying to focus a lot in terms of redesigning the agricultural production system, going much beyond increasing efficiency or substituting the alternatives. So the redesigning is not just focusing on one or two elements in agricultural production. You look at the whole system. So you try to address the agricultural production, the human health associated with the agricultural production, the environmental health associated with the agricultural production. So we look at the production system as a holistic way. So we completely redesign the agricultural production system. So we make the production system to be more resilient than in the past. So now many of our focus is going to the redesigning of the agricultural ecosystem. So all these three systems predominantly focus on the production side at the farm level. So in the new concept, two other levels have been proposed where we are also trying to link the producers, the farmers, with the consumers. So if I am a farmer, when I start producing a rice crop or a vegetable crop, I also keep a consideration for the consumer. For example, if the consumer is expecting a very healthy, chemical-free, nutritious vegetable, then I modify the practices which I follow in the past in order to meet the expectations and demand of the consumer. So this is where we are trying to link the producers with the consumers. And the level 5 is even broader than what we think at the farm level or linking the farm level to the consumer level. Here comes a broader focus of the global changes happening at the several institutions level, including the policy changes which support the development of the new agricultural production system. The good example is institutionalizing the organic agricultural systems, institutionalizing the labeling, traceability systems for the agricultural produce. Okay. So when we talk about the transformation of the food system, primarily it starts with the technological transformation. So we develop the new technological innovations, new technologies, which lead to the changes in the institutional level, meaning institutions engaged in production, institution engaged in the processing, institution engaged in the food distribution, institution engaged in the policy making. So all this institutional transformation is triggered by the technological transformation. And both these transformation ultimately make the broader level systems transformation. When I mentioned the systems transformation, I meant the food system transformation as an ultimate change happening in the world. So how one can contribute for all these system transformation? Push, pull, policy. What I meant to push here is the agricultural production and supply of the food. What I meant to pull here, the consumer who are driving the production systems. What I need, I force the farmers to produce for my need. So that is the pull drive. Then the very favorable policy environment being created. Okay. So when we talk about the food security, immediately we think of some of the predominant crops which we rely on currently. For example, when I say food crop, immediately you may think about rice, wheat, and corn. Probably the young generation may not be aware of any other food grains beyond these three crops. Similarly, when I talk about vegetables, immediately you may think tomatoes, eggplants, peppers, onions, carrots, beans, cabbage. What beyond them? But in reality, there are hundreds of plant species. If you take the leafy greens alone in any particular region, you can see at least 15 to 20 different species. So all these crops make a broad level of biodiversity, and surprisingly, many of these crops are having several fold higher nutrition than the most popular crops what we are eating today. So here at World Vegetable Center, we have the collection of world's largest vegetable germplasm. You will be surprised to hear we have a collection of 456 plant species covering all sorts of major and traditional vegetables, 62,000 different vegetable germplasm accessions collected from different corners of the world. And they were all stored here. We consider it as a treasure box. And this collection is not a museum. They are not the dead specimens. They are active collections. So the seeds of all these 62,000 accessions are regenerated from time to time in order to ensure their viability. And then we use them for further improvement of the germplasm. Not only by our own colleagues, but also we share it with whoever demand it from the different parts of the world. Just I like to show few examples how we use these germplasm for developing improved varieties. You know, when I say about improved varieties, first focus was on improving the yield. So we can increase the yield of a tomato from 20 tons per hectare to 80 or 90 tons per hectare. So high yielding. And most important trait is its tolerance to abiotic stresses. You name it, whatever. It could be flooding. It could be drought. It could be heat. Look at this tomato, wild tomato plant. In a very, very dry land, the soil has cracked like anything, but you see the greenness and the flower being produced by this tomato plant. It has such a wonderful trait of drought tolerance. It can be moved to a cultivated tomato to develop an improved tomato variety. So the tolerance to abiotic stress is there. The resistant genes for the biotic stresses, it could be an insect pest, it could be a disease. You can find a resistant gene in this germplasm. And it's not only yield, not only the stress or the tolerance or the resistance to the biotic or abiotic stress, you can also improve the nutrition of your vegetable. The best example, we call it as a golden tomato, which is a orange colored tomato, which has a 3 to 6 times higher beta carotene, which is the provitamin A, than any other normal tomatoes. So if you take just one tomato, it may be sufficient to meet your vitamin A requirement of that particular day. So we use our vegetable germplasm to develop improved varieties with the unique traits like all these attributes I mentioned here. The other focus that we pay more attention is producing the vegetable crops in a much safer way. Probably you might be aware the vegetable crops are prone to lots of pests and diseases. So the farmers, in an attempt to produce damage-free produces, they apply a lot of chemical pesticides. So we want to reduce the use of chemical pesticides through multiple ways. So I talked about the use of pesticides. You may not believe that the pesticides may be applied like every week or 2 times in a week, or for some crops, almost like every alternate day. So you can imagine how much of the pesticide residues are being left in the harvested produce when it comes to our kitchen, what kind of chemicals are staying, how much impact it could have on the health of the households. It's not only in Asia, but also in Africa, it's the same scenario. This is just a simple indication of the used pesticide containers in the borders of a farm. So considering the time, I'm going to share just 2 quick examples of success stories. One come from South Asia with India and Bangladesh. Both the countries produce a large amount of brinjal or eggplant in the world. But because of the pest problem, the farmers apply a lot of pesticides. Imagine in a 180 days crop, they apply almost 140 times. So you may not want to eat a brinjal with such a pesticide use. So we try to develop a technology which can substitute for the chemical pesticides. So our research and development goes in 3 phases, starting from a technology development, taking it to the farmer's field, and then promoting it to the farmers. So we work with a different range of partners, starting with the research partners, but we do work with the development partners. So here in this technology, it is called integrated pest management. So here it has a combination of different technologies. Some of them are a simple cultural practice, like you cut and destroy the infested, pest-infested plant part, or just you stop using the chemical pesticides. So you provide a way for the natural enemies or the friendly insects staying in the field. Then you provide some minimum intervention, like introducing the pheromone traps, which can attract the male insects. So you attract and kill all the male insects so that you prevent the mating process, so the population buildup can be reduced. So once we did extensive research and develop all these components into a package, we go to the farmers. We use the different means like the farmers training. We use the news articles in local languages, or we make the campaigns, or we produce simple, the extension literature. So through different means, we drive home the message of the new technology. Okay, now you develop a solution. Now you introduce this solution to the farmers. But we are not a commercial company. We cannot make available this technology. So what is missing between us and the farmers is the connection. This connection can be made only by the private sector. So we want to introduce this technology to the private sector. So we provide all the technology at free of cost. The main reason for that is the private sector then can make available the technology for a cheaper price. So the poor farmers are able to afford this particular technology. So in this philosophy, we introduced this technology in India through the private sector, which has reached ultimately the farmers within a brief period of time. And similarly, we worked with the institutions in Bangladesh, but in Bangladesh, we happened to change the existing policy, which provided only the platform for the chemical pesticide. So here comes the second level. I talked about the technological transformation. The second one is the institutional transformation in the food system. So we changed the policy institutions in Bangladesh, which took some time, but after 5 years, we are able to make available the technology commercially in Bangladesh. You will be surprised if you look at the statistics on pesticide consumption in the last 10 years in Bangladesh, you can see a steep decrease in terms of pesticide use in agricultural production in Bangladesh. So we sow the seeds 20 years ago, and today you can see the impact. I don't want to get into the details. Here you can see the impact of the technology adapters through reduced use of pesticide, reduced cost of production, better yield, so better income. So similar impact has been documented even in India. So is the reduced chemical or the increased yield or increased income the only benefit through these technological innovations? No. You can also see the increased biodiversity in the crop production system. So this is a 2-year scientific data accumulated from Bangladesh, where you can see after introducing this technology, the friendly insect population has increased to the level of 50%, starting from less than 20%. So this is called the holistic food system transformation, where you not only focus on improving your yield or increasing your income, you also focus on improving the health of the ecosystem. The second success story is related to the legume, which is an important protein source for the poor household who will not be able to afford the animal protein. So the legumes are very important crop in South and Southeast Asia, as well as in Sub-Saharan Africa. But the legumes are challenged by multiple insect pests, and the pod borer is one of the major problem. And again, the pesticide use is very intensive in both Asia and Africa. So here we come up with a combination of the friendly insects to kill the pest, and some of the microbials which can be developed as microbial pesticides based on fungi or based on bacteria or virus, or even the pheromones. And all these friendly insects are not present in Africa. They were present only in Asia. Similarly, some of the microbial pesticides were not present in Africa. They are present only in in Asia. So we did a lot of research, and eventually we introduced all the friendly insects and the microbial pesticides into Africa. You will be surprised. It has been taken up in a much broader way in several countries in West Africa, for example. And it has been proven very effective in terms of controlling the pest and producing better yield. So the farmers are able to harvest more legumes, and they are able to consume more legumes. But again, how to make available the technology? It is not like in Asia. In Asia, we have a well-developed private sector. So as I showed in case of India or Bangladesh, we easily work with the private sector and make available the technology which can be easily provided to the farmers. But in Africa, there is a very less participation of the private sector. So how to overcome the situation? So we try to identify the social enterprises who are coming up. So we try to work with them, and these social enterprises work with the women help groups, self-help groups, or the women communities. For example, the women communities, they collected 80 tons of neem seeds to produce the neem oil and the neem cake, which can be used as the soil organic amendment. The neem oil can be used as a pesticide. So we work with these social enterprises, and we try to provide the training for them how to produce the microbial pesticides by these self-help groups. And then eventually we make available these microbial pesticides or the neem pesticides to the farming communities. So again, this is a huge transformation which cannot happen overnight, which needs the participation of multiple institutions, starting from the government, starting from the private sector, starting from the farming community, starting from the non-governmental organizations and international organizations like World Vegetable Center. So I talk about how to improve the varieties, how to improve the safe vegetable production, but the story doesn't stop there. You will be shocked to hear up to 40% of the vegetables what we produce in the farms do not reach our plates. They are wasted. They are lost from the harvest in the farm and during the transportation and during the storage and during the distribution in the shops. You go to any wet market like Ulavar Sandai, you go to any retail markets, the big supermarkets, you may see the nice display of the vegetables, but equally there is about like 40% of the fruits and vegetables are rotten. They are wasted before reaching the consumers. So we put a lot of effort and input to produce the crop, and if they do not reach the plates of the consumer, how much effort and inputs are wasted, and how much of the contribution to the greenhouse gas is increased because of the waste coming from the food. So here we also try to develop the technologies to reduce the food losses happening during the post-harvest. This is a simple example. This is called the zero energy cold chambers. This is nothing but the two brick walls, and you put the sand between the brick walls and moist the sand, the fruits and vegetables inside the chamber and close it. And the fruits and vegetables stay fresh for another couple of days to a week. So you can reduce the losses. Solar dryers. So solar dryers can be used to dry the fruits and vegetables. You can store it for a longer time, and then you can consume it during the lean season when the vegetables cannot or fruits cannot be produced or cannot be purchased from the market. So all these simple post-harvest technologies can prolong the shelf life, can retain the nutrients, and can provide the foodstuff during the period of less supply. So we focus on the post-harvest as well. But finally, it's equally important to raise awareness among the consumers to eat more fruits and vegetables. I'm not sure how many of you are aware of the recommended quantity of fruit and vegetable to be consumed in a day. According to World Health Organization and the Food and Agriculture Organization of the United Nations, one must eat 400 grams of fruits and vegetables in a day. I'm sure at least half of you or more than half of you do not eat sufficient quantity of fruits and vegetables. So it's more important to raise the awareness. So that's the reason we start working with even the school children to raise awareness, to introduce the vegetable cultivation through school gardens, community gardens, and then we eventually start promoting more consumption of vegetables. Finally, vegetable production also generating more employment opportunities. If you look at the villages in any developing country today, including India, half of the villages are empty. Most of the villages are having only the old generation living there. The young people are migrating, as I indicated in the very beginning, moving away from agriculture, migrating to the cities in search of jobs. So they try to find employment in the cities. But if you go back to the agriculture, if you go back to crops like vegetables or fruits, they provide more job opportunities. So if you cultivate the 1 hectare of land with rice, if you cultivate the same 1 hectare of land, you happen to spend more man days than the rice crop. So it means you get more employment opportunities. It's not only with the cultivation. In terms of when you market the fruits or vegetables, you get the employment opportunity. And also in terms of economic returns, you get significantly higher economic returns per unit area when you cultivate fruits and vegetables. So vegetables not only provide the food for malnourished population, but it's also alleviating the poverty because of its high potential for better income. So to conclude my talk, for vegetable production and consumption to contribute the food and nutritional security, we are predominantly focusing on the technological innovations to develop high yielding, climate smart, nutritious varieties. We are developing the technologies for sustainable agriculture so that we reduce the reliance on external inputs. Then once we produce the food, we also try to introduce the technologies to reduce the food losses after harvesting. Then eventually we try to raise the awareness among the consumers so that we are creating more demand for the safe vegetables and fruits. And through vegetable production and marketing, we are trying to generate more employment opportunities and income opportunities for the farmers, for the traders, and for the retailers. And finally, we are trying to raise awareness among the consumers to go for more consumption of fruit and vegetable for a healthy lifestyle so that they can remain productive and healthy for a longer period of time. With this, I like to conclude my talk, and I would be happy to take up any questions if you have. Thank you very much.
Thank you, sir. Thank you so much, sir. Participants, do you have any questions?
Sir, good afternoon, sir.
Good afternoon.
Hello. Sir, in this present situation, COVID-19 has affected the entire agri and food system. No, sir. So, what would you prefer the best agricultural policy to accelerate the recovery process that ensures the future food system that is sustainable, sir?
Okay, thank you very much. This is an excellent question, I would say. As you rightly pointed out, the COVID-19 has disrupted the agricultural production system as well as the agricultural supply. So in one side, the farmers produce the crop, but they are not able to take it to the market. On the other side, you see lots of consumers, they are in the need of agricultural produce, but they do not have the access. But, you know, we firmly believe in evolution. Not only evolution of the biotic creatures, but also evolution of the systems. So because of the disruption, do you know how the system has evolved? In countries like India, because of the advancement in the information and communication technologies, every component in agricultural production and distribution try to take advantage of the digital technologies. So the farmers are able to quickly find the market opportunities through some of the digital platform. For example, in many countries, probably you might be knowing about these service providers who distribute the food from the restaurants to the consumers. So the same chain has been used by the consumers and the farmers for distributing the vegetables as well, or fruits, or other commodities as well. So this is called shortening of the value chain using the digital platform. This has quickly evolved in many countries during the COVID-19 situation. I'm sure such a change will also last even after the COVID-19 situation, but the key is we need to have an enabling policy environment. So the government has to come up with a supporting policy environment so that the similar evolutions can be sustained post-COVID-19 era, and the farmers can also take advantage of the same system for procuring quality inputs for their agricultural production. So those sort of changes need to happen so that all the evolutions will become sustainable post COVID-19.
Thank you so much, sir.
Thank you, sir. Thank you, Santina. Any other queries? Any other queries? Thank you so much, sir. On behalf of everyone, I would like to thank you. You told us so much, and especially about how we can develop local biopesticides and also about post-harvesting techniques, because now in the present situation, we are able to cultivate them and also harvest, but post-harvesting is becoming very, very difficult. And you have told us about that also, sir. It has been very interesting for our students. Thank you so much for joining us, sir. And our organizing secretary would like to thank you.
Thank you so
also.
much, sir. Thank you so much for your valuable information related to world vegetable status. And we have learned many things. Surely we enjoyed your session, sir. Thank you, sir.
Thank you. You're welcome.
Thank you so much, sir.
Yeah.
On behalf of all, we would like to once again thank Subramaniam Perumal, sir. Sir, you have been a great support for us. Maybe we should tell, without you, this conference wouldn't have been possible, sir. Thank you so much for all your help, sir.
Okay, catch you tomorrow morning, Dr. Raja.
Yes, sir.
Okay, fine, I'm leaving.
Okay.
Thank you, sir.
Thank you. Thank you.
We are now moving to the paper presentation session. As we already indicated, there will be 4 batches, and you will be given the Google Meet ID in the PDF that we have sent to you. There's a small correction in it, so kindly please note that. Students in batch 1, participants in batch 1. Participants in batch 1 alone, please note the change. Numbers from BB1 to BB5 will be shifted to batch 2. Numbers from BB1 to BB5 will be shifted to batch 2. So please join in the address that has been given in batch 2. And then numbers from BB6 to BB10. BB6 to BB10, kindly join in the link that has been given for batch 3. So once again, I'm repeating. From BB1 to BB5, join in the link of batch 2. BB6 to BB10, join in the batch of 3. And already if your names are in batch 2, 3 and 4, there will not be any changes. Participants, if you have any queries, you can let us know now. You can leave this session. If you're very clear, you can leave this session and join with the Google Meet link. So in case you have any queries, kindly ask us now.
Excuse me, ma'am.
Yes, ma'am, tell me.
Yeah, so batch 2 starts with BB1, right?
BB11, ma'am. Batch 2 starts with BB11.
11, okay.
Yes, ma'am. BB11 to BB20. Along with them, participants in BB6 to BB10 must also join in batch 2.
Okay, so it starts with BB11 as it is in the schedule.
Yes, ma'am. There's no change in batch 2 or 3, ma'am.
Okay.
Yes, ma'am.
Okay,
Thank
thank
you.
you.
Yes. Any other queries? Participants, if you have any queries, no issues, you can let us know now. If you're finding any difficulty to join the meet, kindly let us know. If you're presenting, kindly leave this session and join the Google Meet link that has been given for your particular batch. Students in batch 1, BB1 to BB5, join in link for batch 2, and BB6 to BB10, please join in batch 3. Students who are not presenting any papers today can leave the session. We are done for today and we'll meet you back tomorrow by 9:30 in the morning. Thank you.